All right, here we go. We are live for the debate you've all been waiting for. Now, before we get into introductions for tonight's debate, I do want to welcome everybody to Standing for Truth. My name is Donny, and I am your host and moderator for tonight's much anticipated debate. One could say this is Clash of the Titans. Mark Reid and Matt Nailor, who will be debating the question, is there scientific evidence for evolution? Matt and Mark are no strangers to the topic of creation and evolution. They're both very knowledgeable on this topic and always professional during these formal debates. And so I really do appreciate them both for giving us their time for this epic debate on evolution. So why don't we kind of get acquainted with our guests a little bit before we get into the fun? And so why don't we start with Mark? Why don't we start with you? Great to have you back with us. I think you were here a week or two ago debating David McQueen. So how you been?
Yeah, good. Yeah, busy, but it's always good to be here. And thank you for having me on, and thank you to giving me your time. My name's Mark Reid. I'm an atheist, secular humanist, counter-apologist. I do a lot of stuff on just atheism and secular humanism in general, and I love to debate. So it'll be really good to debate Matt for the first time, and thank you for being here, Matt. Apart from that, I do have some videos going down and some debates coming up. So if you do like debating and you are an atheist in the chat, I'm sorry, but you subscribe to my channel and see all the greatest debates from yours truly.
Very cool. Mark, appreciate the introduction. If anybody wants to see more from Mark Reid and check out all the debates and awesome discussions he is having over on his YouTube channel, please do check the description box for the relevant link. And before I hand it over to you, Matt, Guitar Tunings, appreciate the support, says excited for the debate. Yes, I am pumped for this as well. Matt and Mark, this is their first time interacting, and so I think this is going to be a really, really great debate. So, Matt, you were also here debating not too long ago, probably going back on a couple weeks now, debated the age of the Earth, same topic that Mark also debated recently, and so great to have you back. How you doing? And also a little bit about yourself.
I'm doing good. As you guys know, I'm not too much into debating. I kind of leave that ball in your court, but I run the Young Earth Creation channel, which is the secondary backup channel for Standing for Truth, because we had always suspected your channel to get, into some kind of trouble by now and removed. So mine was the backup channel, and now it's actually doing quite good. It's actually just another channel entirely different than this one. So I don't do much debating. I kind of leave that up to you. But I like to get in there sometimes. This is the first time I've ever debated on this subject, and it's actually very unique perspective because I've always been on the defense. I've never gone against the subject like this. So it's interesting. I'm usually going first and I'm defending a position. So it's different. I guess I'll like it. We'll find out.
Very cool, Matt. Thank you for the introduction. Yes, I'm excited for this. And I'm really excited to see what you both are going to bring to the table in terms of points and arguments. And so with the introductions out of the way, again, I appreciate it, Mark and Matt. I will go over the format for tonight. And so we're going to be having a relatively formal debate. We'll be having 12-minute opening statements. The title, again, for tonight is there scientific evidence for evolution? Therefore, Mark Reid will be starting us off with his 12-minute opening statement. Then we're going to have 8-minute uninterrupted rebuttals, followed by an open discussion. Rather than being a real strict cross-exam, it'll be more free-flowing and organic for about 50 minutes. Then we're going to have a 5-minute closing statement where the debaters can wrap up their thoughts and points for tonight. And then this is where we get you guys in the audience involved. And so we'll be interacting with your questions. The debaters will be engaging the questions that you have for them. And so please make sure you're tagging me either at Donny, and then just let me know who the question is for, Matt or Mark. And so with that, let's just get right into the opening statements. And so, Mark, whenever you're ready, you just let me know. If you need to screen share as well, I can get that up for you. And I see it right now.
No problem at all. Thank you so much. Just let me know when I'm on.
You're good to go, Mark. Floor is yours.
Fantastic. Thank you. My name is Mark Reid, and I'll be presenting some of the evidence for evolution. First off, I do apologize if I go through some things as a groundwork and you've seen it before, but I do think it is necessary to lay foundations for what we are talking about here. So science, you may have seen this before, it's methodological naturalism. It's a systematic field that builds and organizes knowledge in form of testable explanations and predictions. And I really want to focus on testable explanations and predictions because I think that is where evolution really has a strong point in the predictions they make and those predictions being found to be true. So the next question is, what's scientific evidence? And it should support or counter the hypothesis, in this case, the hypothesis that life evolved from a common ancestor. And that sort of brings us to what we count as evolution. Now, there's a couple of definitions here, mostly from leaders in the field. So basically, at a base level, it's a change of heritable characteristics over time, and it only addresses the diversity of life. Evolution is a fact. Almost everybody agrees that some form of evolution happens, whether that mean microevolution, which isn't any different than macroevolution, by the way. It just means over a shorter timescale. And the theory of evolution is the model that describes the processes. And scientists are certain about the theory of evolution as they are about the theory of gravity or the germ theory of disease. As Stephen Jay Gould put it, sort of evolution is both a fact and a theory. There's no competing scientific theory for evolution. Creation science does not meet the criteria to be a science and is instead classified as a pseudoscience. As I pointed out in my last debate, it just does not meet the qualifications, and so there is no competing hypothesis for evolution in scientific circles. Now, to start to go through the evidences, there's tons and tons of them. They all lead us to the same place. This is called multiple lines of evidences that basically end up at the same answer. Now, there are way more than what's on the board. I realize that I couldn't fit them all in, but these multiple converging lines of evidence pointing to the same answer is really what I want to stress on at the start of this presentation. All of this has been unsuccessfully attempted to be explained away and dismissed by religious fundamentalists without success. I don't have time to cover all of them in this debate, so let's focus on what I think is the strongest evidence. Keep in mind, though, there is much, much more evidence than what I'll go into today, and it all leads us to exactly the same answer. All of these evidences support one another. Now, we're going to focus on genetics and paleontology, what I think are the two strongest prongs of the evolutionary evidences. I also want to focus on, as I said before, novel testable predictions. Novel testable predictions are what we expect when, or what we find when something is not known or unconsidered, and we make a prediction and that turns out to be true. There are so many of these associated with evolution, but I'll at least give at least one for sort of each line of evidence that I'm presenting, at least one. So the first off is the fossil record. Now, fossils provide solid evidence that organisms from the past are not the same as those found today. Fossils show a progression of evolution through the strata as we go down. Fossils, along with the comparative anatomy of present-day organisms, constitute the morphological or the anatomical record of how things have changed over time. And don't forget that evolution is simply change in heritable characteristics over time. By comparing the anatomies of both, we can infer lineages to species. Now, that approach is the most successful when they have hard body parts like bones or shells or teeth. But we can always predict to find basal species, i.e. less complicated species and body patterns and morphology, lower in the strata than modern species. And this prediction, when we go out to find out whether it's true, always keeps coming true. There are also a couple of real notable predictions in the fossil record. This is Archaeopteryx. It was uncovered in the Jurassic limestone of Bavaria just 2 years after the publication of Darwin's Origin of Species. It appears to be one of Darwin's predicted missing links. In this case, the link between reptiles and birds, specifically between dinosaurs and birds. Over the years, 12 body fossil specimens of Archaeopteryx have been found, and that is a successful prediction made by Darwin. Hilariously, in 2007, Ray Comfort and Kirk Cameron asked to see a crocodile, saying that it should exist if evolution is true. Again, like Darwin, 2 years later in 2009, a fossil of a mosaic species called Anatosuchus, literally called ducked crocodile, was found that had a crocodile-like body but a broad, flat duck-like snout. So well done on that prediction, creationists. Here's another example of where evolutionary theory can help us find things and demonstrate that the model is in fact true. This fossil was specifically looked for by researchers. If evolution was true, they should find an intermediate form between fish and tetrapods. They went to the stratal layer that they predicted and found Tiktaalik exactly where they had predicted it. And when I say exactly, we're talking in sort of millions of years, so sort of a 20 million year period. But it did occur exactly when they predicted it on a geological scale. The other thing is genetics. So let's go for the retrovirus elements. And you may have seen this before. I do apologize if I've gone off before, but I'll go through a quick explanation. If you do want more information on these, I have done debates on them in the past where I went into a much further detail. So a retrovirus is a RNA that has, instead of DNA as a genetic material, it uses an enzyme to make itself into DNA and another enzyme to make a cut in the host, and then insert its own DNA sequence into the DNA of the host. This makes the host replicate the virus and then transcribes the DNA sequence as a sequence now included in the retroviral DNA sequence. So basically, it inserts itself into your DNA and then keeps using you to replicate itself. An example of this is HIV. Now, the structure is very unique of these things, and long terminal repeats on either side, GAG, POL, and ENV basically show that these things are viruses. So how does this make this evidence for evolution? Well, basically, we have these integrated into our DNA in exactly the same place. And in fact, we can see that crosses over different species. For instance, we share something like 208 of these with chimpanzees. Now, if they were, these things have to, because they're in every single cell in the body, they have to be an infection of a germline cell, either a sperm or an egg. That is the only way they can get into every single cell in exactly the same place in our body. So that shows that we must have had a common ancestor whose germline cell was infected by a retrovirus. And we know this for sure. And we also know they're retroviruses and not some sort of other genetic sequence. And I'll show you that later. What I want to look at is how we can co-opt these retroviruses to help. And a big indicator of this is the cytosine family. But this shows that it's not necessary to have cytosine in order to reproduce. The marsupials don't use it. The monotremes don't use it and still lay eggs. So the SIRH family is present through all placental mammals, but not in monotremes and not in marsupials. Surely you would think that a common designer would use the same sort of design in order to facilitate offspring, but no, apparently not. So it's not necessary, but it is of great benefit to placental mammals. Now, how do we know these are definitely viruses? This is the reason why we know that these are absolutely viruses without a shadow of a doubt. Now, this is HERVK. It is the most recent human endogenous retrovirus, so the youngest that has been inserted into the human gene line, and it is the least mutated of the viruses that have been inserted. Now, this is basically the budding that occurs when they reactivated the virus. So it's a reactivation of the HERVK virus that has been long since dormant. It shows that sort of in vitro recombinations along with present human day HERVK can generate functional HERVK elements. So it can generate functional viral elements, including it can infect other human cells in vitro. In vitro. The virus turned out to be weaker than expected, but that is completely what they predicted. So the prediction is that if these things are in fact viruses, they should be able to reactivate them into an active virus, and that prediction came true. Now, the other one is chromosome 2 fusion. Since we have 2 less chromosomes or 1 less pair than the other apes, it's predicted we will find the unique signature of chromosome fusion in the genome of humans. The chromosome 2 fusion site is exactly what was predicted, and it shows beyond a shadow of a doubt that human apes had a common ancestor. The telomeres are highly replicated groups of sequences that regulate duplication. Now, these get shorter if the cell replicates. So the prediction is that we should see high density duplication, yet degraded sequences of head-to-head series in the middle of chromosome 2, but they definitely should not be. And the reason why they should be highly degraded due to the fusion having to happen, various deletions has to occur when the mutation happened, making the sequence degraded. And we found exactly what we predicted, showing that once again, evolution is true. And that sequence, by the way, is about 798 bases. So it has been said that there's other sites that have this repeat. Just for your sort of information, those sites only have usually 2 to 3 repeats in them, while this has got 798 repeats. So this large amount of duplications is what we should see when we actually see telomeres. And the fact that it is degenerate, I mean, if it was pristine, it would just be a telomere. So the fact that it is degenerate, that is what was predicted, and that is what we found. So all of these are novel, testable predictions that evolution has made that have actually been successfully fulfilled, as well as tons of other ones. There's no end to them. Yeah, I think that will do it, and I thank you for your time.
Mark, it's like you've debated before. That was literally 12 minutes on the dot. So great job. Appreciate it. And we're now going to hand it to Matt. Matt, before I do, I want to let the audience know there are a lot of questions coming in. So that's great. I appreciate how engaged the audience is. And so I'll let you know I am all caught up, and I appreciate it. Okay, Matt, whenever you're ready, you just let me know. And you also have 12 minutes. If you need to screen share, go ahead and just let me know when to start your timer.
Okay. Yeah, I'm ready to go.
Right, you're good to go. Starting now.
All right. I have no doubt that Mark believes that the evidence for evolution that we have portrayed in the textbooks is proof for deep evolutionary history, just like I think Mark believes the same about me regarding my interpretations of the evidence for creation. So when it comes to that, Mark says he likes testable predictions. Well, that's great, because ironically enough, that's what I believe is one of the strongest points for young Earth creation. So he might not know these, so we're going to go through some of them. One of these is the most important genetic prediction of all, which is genetic similarity. This was not predicted by evolution. That was actually predicted by a young Earth creationist. It was in the 1960s that evolution actually predicted the exact opposite. It was Ernest Mayr who said, we will not find genetically similar ancestors the farther back we go in time, because genetic mutations would have scrambled it beyond recognition, and we won't find any genetic similarities. It was the young Earth creationist in 1975, the Institute for Creation researcher himself, who owned it, who said, no, actually, we would find a genetic similarity in all life, because he asserted that there would be a common underlying designer that would have left a pattern in all life, not showing that there would have been multiple creators. Otherwise, it would look like there were, maybe if you still want to believe in God, that there would have been multiple creators, right? And to the astonishment of the evolutionary community, no biologist had even the foggiest notion that such similarities could exist between genes of different animals. So it was a perplexing thing that actually went against evolution. Ironically, now they use it as the best evidence for evolution when it was an actual falsification of the theory. So they had to backpedal and say, no, now they actually use it, when in reality, it was completely against them down the line. So obviously they had a retrodiction, and they use it for their styles now. So I like to say, the model does make really good testable predictions. This list is the ones that just have to do with genetics. And this is where we really, really, really shine. And I would say, what makes more sense? We have, let me try to find my pictures. I don't see one on that. So I would say what makes more sense? About 200 to 300,000 years ago, primitive man goes into a bottleneck, and modern man, there we go, modern man comes out the other side. Then remains in Africa for 150,000 years before venturing out and then evolving into the European and the Asian people groups. Or does it make more sense that humanity arose and mostly diversified from the Fertile Crescent, kind of by Egypt, that was working together to power and diversified and filled the earth, which means that phenotypic diversity is the same between all different peoples that are in the world today, which is exactly what we find. We not only find that mutation rates point to this generation of Peleg, but that all of the haplogroups themselves explode from this very region of the earth, not in Africa. Why is that? Then we find the Tower of Babel. If Africa was really the home for all the people, we would find a nested hierarchy where Africans have the most phenotypic diversity. But instead, when we look at people, it's exactly the same everywhere we look. And then we go over to Africa, we find very few haplogroups. That's very strange for a lot of people groups that lived in small populations on a continent where the temperature and demographics and latitude, longitude vary more than anywhere else on Earth. You think these mutations would have been rising just like they do today and everywhere else in the world rapidly, but we didn't see that. Everything points to people arising just 5,000 years ago. You can go down the line. It's just mutations. We can go to dendrochronology. We go to desertification of the things. We go helium death rates. We go books and history and population growths. We have genealogies that go all the way back. We have history itself going back to about 5,000 years. We have mathematics, language, writing systems, ice age, species, going back to the same thing. It's very unique that everything seems to point to that. But how about this also? What makes more sense then? Is it true that 9,999 other women all just only had boys or just left their lineages and there's no evidence for them at all? That really just doesn't make much sense, but yet we go to the mitochondrial mutation rates. They go back to a mitochondrial Eve just thousands of years ago. Why would it land on the young Earth creation timeline? Why can't it go back to even 100,000 years? You can't find a single pedigree study that does. What about genetic boundaries? Probably the most important thing we can talk about, because evolution says there are no boundaries. Microevolution leads to macroevolution, but yet that's not what we see. We see these genetic boundaries, and we have studies now that are revealing this, and they admit that was totally unexpected. So again, another non-prediction from evolution. They predicted the opposite, which in between species should be arising all the time, but we're not really seeing this. We see independent lines of the family going back. The authors of the study from the 2018 Rockefeller University study said, this conclusion was very surprising, and I fought against it as hard as I could. Why? It's because it's a mindset thing. We were all taught, Mark, all of us were taught evolution is true. So when conflicting evidence comes in and it goes against that, you're like, well, evolution is still true, but what can explain this? Well, that's the problem. If you're only allowed to accept evolution as the only answer, then you're going to have a problem. Anyway, I digress. 90% animal life, genetically speaking, rose at the same time, including human beings. That means fish did as well. They went through this global bottleneck. And they admit, this debunks the notions of what we thought about evolution. This completely contradicts it. Matter of fact, what it does show is confirmation of the biblical creation model, because we say that there was a global bottleneck and that 90% of all life died and 10% made it on the ark and got off and populated again. So I say the more we learn about the science and the more these predictions keep coming true, I think it's going to lean back to the young Earth creation model as being the strongest one. As you see, they expected many more differences based on deep time being true in the human population. That wasn't the case. Matter of fact, when we looked at DNA barcoding, which classifies life, they said this discovery confounds predictions from population geneticists about the theory that older or younger populations should show more diversity. So it conflicts with evolution. So what did they do? They went to their fossil record. Well, that was a problem too, because when they went back 200,000 years in their version of the fossil record, which is a picture of time, they found nothing. Why? Because the fossil record isn't an accurate depiction of time. It's an accurate picture of events that happened, not time. So I say that's why these genetic barcoding shows so much genetic similarity in time. Why? Because there's a fast mutation rate and there's not a lot of diversity. You can't have those two things happening without young Earth creation. It just doesn't work. Instead, we find individual lineages going back to a bottleneck, not a convergence on common ancestors like they expected to find. We find exactly what you would expect to find in the biblical creation model, which talks about these different organisms having different families, right? And this is what's going on. So another important thing is if time was really, really old and lots of time had passed, well, the mitochondria accumulates mutations as time goes on. Dr. Nathaniel Jensen said if the amount of mutations that we're finding keep compiling, we should find about 21,457 mutations in the mitochondria, which would be a total saturation of 12,000 before we couldn't see anymore. We do find more. We don't find anywhere near that. We find under 2,000. Why is that true? If deep time is true, if even 200,000 years had passed, the mitochondria would be riddled with mutations, and it's just not. We look inside the CO1 gene, which is a highly conserved gene. We find only 65 mutations in it when we look at primates. We find almost none in human beings. And then, but there should be 466 total mutation saturation. And we know this is an accurate mutation rate because the NIH itself uses the young Earth creation model. They don't use the evolutionary model. They choose Dr. John Sanford's model, which uses the observable mutation pattern of how mutations spread through a population. And they use it to trace who is going to inherit cancer. And the NIH uses it. Why don't they use some evolutionary model if evolution is true? Why are they using the young Earth creation model? It's because it's better. It's actually working. NIH doesn't care about evolution and young Earth creation debating. They're not even going to watch this video. They don't care. They just want to know what works. And what works is the biblical young Earth creation model. And there's books written about this, like Why Aren't We Dead 100 Times Over. They write all these books and they give explanations on trying to figure it out, but they don't have any evidence for it. So here's the thing. The evolutionary theory has become so plastic, it can't be refuted. It's unfalsifiable. He wanted to go to the fossil record. Well, guess what? The fossil record doesn't show only slow gradualism over time. It shows punctuated equilibrium. It shows complete stasis. It shows de-evolution going backwards. There's Lazarus taxes. That don't make any sense at all. Like they die, they vanish for hundreds of millions of years and they reappear again, completely unchanged. And then there's ghost lineages like cats. They just arise randomly out of nowhere. There's like a giant cat. Where'd they come from? Who knows? So the fossil record is kind of a mess. And they admit that too. Like they go, well, it's kind of a mess. There's no consensus on what's going on. We can get into that though when it's time. Let's see. I just was going on tangents there. So I would say that the theory of evolution to me is a lot like the Histories channel, what they show today, which is ancient aliens. It's true. They find evidence of giant megalithic structures and pyramids. But did aliens actually do them? Of course not. So I think, sure, there's evidence in the ground that we can find of things that have died. So it's the story around those that we need to deal with. And yes, we can tell all kinds of stories with the bones that are found on the ground. But again, the genetic evidence is, to me, the biggest and strongest key. You know, there's almost 20 seasons of Ancient Aliens coming out. So clearly this is very popular. And clearly they have lots of evidence to back up what they think is a really good theory. But to me, it doesn't hold any weight because it doesn't make any testable predictions. And the ones that it has made have been falsified, just like Darwin. You know, let's go over to Darwin. He made tons of predictions, right? He's only gotten one right. And by the way, that was actually something he plagiarized, which was natural selection. He plagiarized from a creationist. He didn't even make it up. It was Edward Blythe who did it, a creationist. But he predicted based on evolution that their finches would arise once every 3,000 years based on evolution timeline. Well, guess what? Recently, we can test that. It's completely wrong. They arise every 2 to 3 generations. That's completely opposite of what he predicted based on evolution being true. They rapidly form, within 2
1 minute.
1 minute? All right. Animals are rapidly forming. This was a creationist prediction. Matter of fact, they are even saying that Darwin could have never even imagined how fast these things are forming. They're talking about needing a new theory of evolution to cope with this and to figure out like what's going on. How can we explain this? We need something better and what's new. So the biologists themselves in the Darwinian theory are saying we need something better. We need something new. And if the theory itself can't even be falsified, that's bad news. You know what I mean? So to me, it's like, well, mutation rates prove it. Well, yeah, when you create your own mutation rate, of course it's going to align. You just invent numbers and throw them out there. If they actually worked, they would be being used. What's being used? The young Earth creationists. The FBI is using the mutation rate by creationists, by Parsons, who found that it's super fast and points to a mitochondrial Eve 6,000 years ago. So anyway, you know me, I can keep going. I'm good. I don't want to throw so much out there he can't answer anyway. So I would say, okay, well, here's one, Mark.
Well, actually, Matt, just to be fair, we'll stop there. That's 12 minutes. So good job to the both of you. Lots of points and arguments on the table to rebut, and then also for us to engage in the 50-minute discussion period. So thank you to Mark and Matt for your 12-minute opening statements. We're now moving into the 8-minute uninterrupted rebuttal portion of tonight's debate. Mark, whenever you're ready, please let me know, and I'll start your timer.
Yeah, sure. That was a lot of scattershot there, but let's get into it. Let's go. Let's do it. Okay, so predictions like this, where they basically come out and say, "Oh, well, we predicted that genetic similarities and stuff." We've got to understand that they don't actually do any scientific work regarding those predictions. How much variation we should see, how much similarity we should see. They just throw out things. Don't forget that creationists, first off, completely denied that evolution was a thing at all. They've been dragged kicking and screaming into the 21st century because as time has gone on and the evidence has mounted and mounted and mounted, they've had to admit there has to be some sort of evolution happening, even if that is at a micro level. So this whole thing about, oh, we predicted similarities, yeah, they threw out a lot of stuff. And occasionally they get something right, but they didn't actually make any significant actual specificity with that prediction, and that's an important point. Genetic mutations, well, I've got to primitive man and modern man and the haplogroups that it went to, because we all have, like, if you're talking about haplogroups as in the ones that have no other haplogroup with them, like non-BT haplogroups, yeah, they just exist in Africa. But the A haplogroup, the whole point is that A diversified at the BT split and went into haplogroup B, and other haplogroups C, D, E going on with, well, D and E is its own split. But the whole point is that forms a nested hierarchy. Obviously, Matt doesn't understand what haplogroups actually mean. And it's the whole point about having 3 different sons create 3 different haplogroups is ridiculous. These splits, they have to be separated genetically for a large amount of time for that haplogroup to split. And there's intermingling back and forth, there's recombination, there's all these kind of things going on, there's genetic drift happening. Yeah, evolution is messy. I'm sorry, it just is. Like, you want it to be neat, and it's just not neat. It's a messy progress where people go back, breed with other people, move away, they're genetically split. Ring species occur where they're split and then can't breed when they come back together. There's a whole load of things going on. So I want to ask Matt if he's aware of the BT split and whether he knows that we all have the genetic sequences from haplogroup A in our Y chromosome. Is he aware of that? Because that just shows that sort of what he's trying to project is not true. And he misunderstands mitochondrial Eve as well and said, oh, all these women, just died out. No, they could have had children. They could have had female children, not just sons. This shows you don't understand how it works, mitochondrial DNA works, because they can have daughters. That's fine. But if the daughters and their daughters die out, or they produce sons, that matriarchal line will end. It's not a case that they didn't have daughters. It's just that those daughters and that mitochondrial DNA didn't make it into present day. That's it. So it's baffling as why you sort of said that. 90% sort of, when you say 90% of the species came at the same time, we're talking in geological eras. So we're talking over millions and millions of years after the sort of extinction. Now that time was 100,000 to 200,000 years ago, and sort of the article that you're referencing brings that up. So, this whole idea that, hey, and it's not like modern animals just appeared out of nowhere. We're talking about modern versions of animals that already existed, like the megafauna that was already around, and other sort of more basal creatures, things that we find in the fossil record. They didn't just spring into existence. They evolved during this time. And that is over a geological timescale, not sort of in the middle of one Sunday, 4 o'clock. When they say at the same time, they're talking relatively, not like absolutely. So the whole idea of a bottleneck, has to be Adam and Eve is ridiculous. We know that the human species has never dipped below 1,000 members at any time. We can prove that genetically. Mutation and genetic entropy. This is absolutely silly. So we use the Mendel's Accountant, I believe it is, by Stanford. But it only has one mutation rate, and that's all. right? So it is useful under certain circumstances. If you want a broad view of populations with a constant mutation rate, it is a useful tool. I will happily admit that. But what it doesn't do is actually predict things that happen in real life. Like, for instance, fruit flies, Drosophila. That has been doing years and years. And according to Stanford's model, they should be completely and permanently beyond repair, but they're not, and neither are the bacteria in the Lenski lab. Neither is anything that we've seen put into place with field experimentations. They don't have this genetic entropy. It is just not borne out in the experiments. And so we know that that just is not true because we have put into place these experiments to try and test it. Now, the whole thing about origin stories not being compatible, yes, we're kind of saying, hey, things got messy when humans originally came from Africa. We know that because of haplogroup A, that because that is the haplogroup that is still in Africa and everything leads on, we know that's where it originated from. Was not the Middle East. No, was not. Definitely not. And yeah, there might have been populations moving away, coming back. And this is what I want to point out. Things get messy. They get incredibly messy because Matt probably thinks of it as this, well, there's one person and then that produces this and it's all very static and it's all very like, just one couple produces this. No, it's absolutely populations moving away, populations coming back, populations recombining, populations like moving away, then coming back, then moving away again. Like there's no simple sort of method to do this. And the whole idea, we have a sort of a fallacious argument, an argument from sort of wishful thinking that, mutation rates shouldn't vary. Why? Because that's convenient for us? Well, I'm sorry, nature doesn't do what's convenient. Mutation rates vary. That is just a fact. And just because you have a model that can be useful when you apply a mutation rate that doesn't vary, doesn't mean that is true, because that model, when put into practice in real life situations, does not bear out. It just doesn't. So sorry, I mean, that's not to say that the model isn't useful. It is. But yeah, I think that's about all. Yeah, and the whole idea that Darwin's theory hasn't been used for ages. We're way past Darwinism. We're onto integrated synthesis, which is a whole bunch of different mechanisms that we are finding work together, kind of thing. So, I don't know why you'd be bringing up why Darwin's theory didn't work, because we know Darwin got things wrong. There were things that Darwin was unaware of. Absolutely, Darwin got things wrong. That's the beauty of science. We learn as we go along, unlike dogma, which never updates at all. And all that Matt can do is find ways to get this sort of fitted into his narrative that he has. And so, yeah, I don't know why Darwin's being brought up at all, really, but sure.
Mark, thank you very much for that 8-minute rebuttal. We're now going to hand it over to Matt. Matt, whenever you're ready. You've also got an 8-minute rebuttal. And so just let me know when you're ready to go and I'll start your timer.
Okay. I guess we're good.
Okay. Floor is yours. Go ahead.
It's very hard when there's a lot of topics. I know what you mean now. Okay, testable predictions. So I know that you don't like how we make the testable predictions because we can't originally test them, I think you were saying. Well, that's pretty much why we make them way ahead of time, even if we can't do them. It's like saying you don't like that Ernest Mayr made a predictions that we shouldn't find genetic similarity in things because he wasn't able to predict them or we weren't able to test them at that time. But that's how it's done. That's actually what makes them unique and novel and descriptive. So just because we can't test them, that doesn't really mean too much. And the detail of it, well, there's only so much detail that can be given. He was just saying, like, he literally said, we won't find genetic diversity because deep time will have erased it. That was pretty much all he said. Mutation rates will scramble any genetic similarities that we will ever find over hundreds of millions of years. Therefore, evolution had to make a retrodiction and say, well, there must have been a bottleneck. So when did they say? About 200,000 years ago. Can they prove it genetically? No. Can they prove it in the fossil record? No. I showed you that. So it's a rescue device is what it is. It's just like, well, we don't have any evidence, but it had to have happened because everything is so genetically similar. But why is it genetically similar? Well, only using evolution, we only have one answer. So that answer becomes, well, evolution had to have been a more recent process through a global bottleneck. Well, that's what we've been saying. That's what we would predict. So is it true that creation has changed over time? Absolutely. When that court case went on, they didn't have even young earth creation. It was intelligent design that was on trial, and they only had predictions on junk DNA, which couldn't even be tested. So now we have tons of it. Just like evolution theory has changed over time, so have we. That's a good thing. You should want us to change because we're scientific. You guys are asking us to give you scientific explanations for biblical creation. Even though the Bible is not a scientific textbook, we're saying, look, if this is the true history of the world, we want to give you scientific answers to show that. So we do, and then it doesn't seem to be good enough, but it's how we want to do it. It's for you guys. It's literally for you. It's creationists don't care. It's the evolutionists that seem to really not like the subject so much. And that's another thing that I don't really understand so much. Evolutionists don't like to be called evolutionists. They say that's not even a word. But yet, if you call a creationist, a Christian a creationist, none of us care. It's like, it's just the way it is. I don't see the problem with it. Classifications. Luckily, science isn't determined in court case, so that's good. You talked about comparative anatomy. Here's the thing with the comparative anatomy. That's actually changed again recently. Hold on, I don't know where I put this. What we're looking at is actually the new studies are actually showing that morphology, that homologies, what they've been using in the fossil record to determine what's related, is actually useless now. The newer studies showing the molecular data is way more efficient. The molecular data is showing that if you base something on the morphological look of something, that it doesn't work anymore. The trees, the evolutionary trees of life, are just falling apart. So they're saying we need to get rid of the homologies. Well, guess what? If homologies are gone, you can't use anatomical similarities as evidence for evolution anymore. And so now you're left with just the genetic evidence. But the genetic evidence is what we're saying are functionally designed elements. So how can I just say that? Well, again, we want to go with what Nathaniel Jensen was saying, right? He made these predictions. That's what evolution predicted. He basically said the opposite. He said, in contrast, the evolutionary model predicts that there's an explanation of molecular unity and diversity. What's going on here? He said that the model holds that the variation that we find in the mitochondrial sequences within families are functionally neutral. And so these predictions are what will run. In 2015, we started to find that to be the case. If we want to go off the phylogenies that are based on evolution, we find weird discrepancies because when we're looking at cytochrome C, we find that pig is more genetically similar to human beings than the orangutan. But that breaks that homology down evolutionarily, right? Because obviously in time-wise, that doesn't make much sense. But in the biblical young earth creation model, we want to look at the similarities inside to determine whether or not we're related genetically and where that border is. So that border shows us that when we trace mutations back, we land on a human being. Just like when you trace it back with a pig, you land on a hog. With a dog, you land on a wolf. That got off the ark and diversified. That's the creation model. So why is there genetic similarities that look like this in aquatic species? It's because these are functionally designed. How can I just say that? Anybody could say it. So when we go to fish, this is inside the CO1, which is a highly conserved region in the mitochondria. When we're looking at fish, we see those little black arrows. But here, let's look at this region that doesn't have any mutations in it. We find that it leads to a fish that has a fully functioning fin in the back. But when a mutation occurs and we find those arrows and we trace them to the fish, what do we find? We find that that function gets disrupted and that they lose their tail and it doesn't function anymore. So we see a dysfunction going on. So we know that there's function going on with the other sequences. And when those sequences are messed up, when we find the mutation in a chimpanzee or a gorilla, for example, here, they get horrible genetic problems. They lose their hearing, they lose their sight, they get neurological problems. In humans, they get all kinds of diseases. But yet they want us to believe that all of those mutations right here are evolution over time. But yet when we get mutations in those regions, they cause nothing but disaster. As a matter of fact, that's what's killing the gorillas today, is the mutations that originate in these regions. So if we're finding that none of these mutations can add any of the genetic information that's going to lead to a higher species to form this slow microevolution over time to a macroevolution, it just can't do it because these mutations are causing horrible problems down the line. And lots of scientists, I don't quote creationists because critics don't want that. So it's just, we're finding problems down the line. You know, we see dogs are very genetically similar to us, but yet when mutations end in the same nucleotide sequences, they get the same disease. So we're finding a lot of the pattern that we're predicting, that these sequences don't just do one function. The CO1 gene is notorious for just having a single function, which is cellular respiration. So when I show people, like, look at this region right here, that's not just doing that. Dr. Nathaniel Jensen predicted that these are going to have secondary and third functions. Well, that's exactly what we see. And we find mutations in them, they cause problems. Well, we shouldn't be finding problems. We should be finding much more benefits coming on, because you can't get evolution if you're making tons of steps backwards.
1 minute left.
And then no steps forward. All right. So a lot of people don't like genetic entropy, but here's all the scientists that all throughout the time have actually been agreeing with it. And you don't get much better in genetics than Dr. John Sanford. And it's what converted him from evolution to being a young earth creationist, is because of the evidence that he found in the genome. And he's really into the subject, as you could imagine. So again, we want to make the testable predictions. Again, we're finding exactly what Nathaniel Jensen predicted. And here's the study where he said, did God create individual members of a kind within the same starting sequences? The results of this study are consistent with that hypothesis. So he made the prediction, he ran the numbers, and we found exactly that. They have multiple functions and they have multiple things. And if we're only seeing mutations cause problems, then that's another one. So why are species more primitive in the bottom of the fossil record? Because when there's a global flood, things are going to be buried in the ocean first. So it just so happens that
5 seconds.
More primitive. Okay. Whoa, no, I didn't answer the ERVs. Oh no, or the chromosome too. I'm sorry, Mark.
That was kind of like a second introduction, not really a rebuttal as such. You didn't really rebut much.
Oh, bring in the heat. Gentlemen, bring in the heat. I like it. Okay, well, I'm going to stop it there. So that is 8 minutes for the both of you on the rebuttals. And so that concludes the openings of 12 minutes and the rebuttals. Lots of points on the table. Good job to the both of you. Fast paced. And I know these rounds fly by, but we've got 50 minutes to discuss each of the relevant points and topics. So Matt did just end with his rebuttal. Therefore, Mark, why don't we give you the floor to pick the first topic for discussion? Go ahead.
Yeah, so I brought up really two main topics: genetics, which was ERVs and sort of chromosome 2 fusion, and the fossil record as my main topics. I noticed that Matt didn't rebut any of that. He just simply said, well, fossils will be buried at the bottom, but that doesn't explain why there are, for instance, dinosaurs always buried beneath birds, for instance. That had nothing to do with a flood. And how did all of those bones get organized? How did the flood organize all of them according to their basal patterns? How did that happen exactly?
Well, you have to remember that if that was true, then the fossil record would be very, very simple. I mean, we would find those gradual things leading to it. Matter of fact, we find birds before dinosaurs now, before the very theropods that they're supposed to come from. These are basal
No, we
birds.
Don't. No, we don't.
Okay, well,
no, we don't.
And so you think the theropod dinosaurs, like the velociraptor, that line led to birds?
Yes. Yeah. So Archaeopteryx is a perfect example of this, as I brought up, that you didn't actually address. It is a theropod dinosaur because it has all of the hallmarks of a theropod. It has unfused wing fingers, but it also has feathers. It also has the breastplate of a bird. So it has the wishbone arrangement. And it also has teeth, even though it has feathers and wings and a bony tail. So it has a lot of the characteristics of birds and a lot of the characteristics of theropod dinosaurs. So how exactly did this happen, and why do we only find that further down in the fossil record, and then you don't get any birds whatsoever below that? So the idea that you find birds before dinosaurs, I'm really sorry, Matt, that just isn't true.
Well, I would say the one thing that you mentioned was that why do we find them in that order? Well, we see mosaics, which would have more things inside of that creature that we don't find today. We believe in de-evolution, right? Something was created and then it only lost information over time. So why would a bird have teeth back then, have the ability to fly and run on the ground? Because they have much more genetic diversity built into them than we find lost today. So we're finding, we like to say that mosaics are always going to have more information, very similar to the giant bat that we found. We look in the fossil record, and the first bat that we find is called a giant bat species. It can both fly from the ground. Most bats have to drop to fly. They can run, they can walk, they can burrow under the ground, they can eat fruit, they can eat insects, they can eat animals. So they're a very good mosaic because they contain all of the characteristics modern bats have in different lineages all together in one. So we would say the same thing with a lot of birds. If we're going to find an ancient bird, we would find that it would have, of course, mosaics, but it had more diversity. So it could lead to the different of some of the different bird species. But we've found in the fossil record lots of different birds. I've mentioned, I can show you different studies that where they find the loon, the parrot, all these things in the fossil record with dinosaurs. Here's one that's specifically talking about the Archaeopteryx, and it says here we highlight one issue that is often considered to raise problems for the theropod hypothesis of avian origins, and that is the temporal paradox. It is this, I hope you can see it, stratigraphic distribution of the theropod fossils. The idea that the earliest known avian is the late Jurassic, but most other groups are poorly known in the Jurassic, implying that the avians arose before their supposed ancestors. That's what I was talking about. We find the bird.
Yeah, so hold on. You're sort of gish galloping along there, Matt. So let me address your points because you're sort of going on and on and on. So the whole thing about de-evolution is you've sort of said, hey, things had more sort of utility kind of thing. But the problem is that you also get sort of sauropod dinosaurs back then, which all they were is just big tanks. All they did was eat, essentially. So all they did was just stand around and move slowly, and they were basically herd animals. Like there were a whole bunch of dinosaurs that only had one very, very niche functionality. So the idea that more adaptability or the ability to have, multiple different functions like flying and burrowing. I'm not familiar with this bat. But a bat that burrows, I would like to see the citation on that one. That could fly and take off from the ground and burrow, I would like to see that. They're also talking about that because when we go back to say, hey, the origin of birds is this point in theropod dinosaurs, see, what you're doing is you're cherry picking from papers. That paper supports evolution. I will guarantee you it will. It's just that you're picking out things from it and misinterpreting them exactly how you want to. You've done this with a few other papers before. That they do say that this creature, because what happens is we find the earliest transitional species between theropods and Archaeopteryx isn't the only one we found, but we find earlier ones and we have to revise our estimate. It's not like we're just going, well, even though we found an earlier one, we're just going to keep the estimate the same. But none of those estimates have any problem with the theropods changing to avian, and the genetics match that as well. There's converging lines of evidence that these theropods did turn into birds. And I noticed that I asked a question of how do you explain the different attributes that the Archaeopteryx had, but you just completely ignored the question. Why does it have the wishbone, teeth, which no modern bird has? That's a theropod teeth. Why does it have a bony tail, which no modern bird has? Why does it have the characteristics of both a theropod and an avian animal?
Well, we also think that a lot of the creatures that were then are going to have more diversity. So dinosaurs are gone, right? We don't have any more of those. So of course we're not going to have dinosaurs because you mentioned like how come there's no you said the dinosaurs are they're herbivore oriented. They're very specific to their niche. They didn't really do very much. Well, we don't have dinosaurs today to go off.
I'm saying some did. Some were very niche and did specific things. They were very good at their specific thing. But you sort of said versatility was more prominent in the past because of this de-evolution idea. But if that were true, you wouldn't have these niche dinosaurs like sauropods, like the big brontosaurus. It just basically ate and it didn't have a lot of this versatility. It was basically just a huge herd animal.
Right. Well, maybe that's why God only created to do that, right? A giant vegetation is going to need a gigantic garbage disposal to digest all of those things. Matter of fact, there's something called the sauropod paradox. They actually believe that they couldn't even walk around because they would have killed most of the vegetation on land. So how could the vegetation have even lived? So maybe God created these things to step on and destroy lots of this vegetation so it wouldn't be such overgrowth. They didn't if they survived, they died out not long after the flood, if they were even taken on the ark to begin with. We don't have to say that they were, because they don't exist today. We can't. So I can't say if brontosauruses lived today and they had the ability to fly, we'd be like, oh, our theory is wrong. They evolved this unique special ability where they gained lots of information and now they can fly around. Well, if they were alive today, I'm guessing they would do the exact same thing that they were doing. They would just be walking around grazing like cattle would be. So we were saying evolution needs to add the information, because you guys are looking at a linear scale on the evolutionary charts showing, basal creatures with small genomes arising into all the diversity of life we see today. We say, no, you can't really get that by looking at the fossil record. That's why they say, no, we're at the tips of the nodes of the evolutionary branches, and we can't see that today. They say that because we don't see any of the evolutionary trees of life.
Okay, so birds didn't evolve from sauropods at all.
No.
The whole idea that sauropods were created to, I've never heard of this sauropod paradox. I don't know where you're getting that from, but sauropods couldn't eat because they would plod and stamp on things and destroy them. I've never heard of this hypothesis before, but I'd like to know how you go about demonstrating this. I've never heard of this before. I think that there might be maybe some merit to they ate so much that plant life would have trouble staying alive. I'm not sure if that's a problem. I think that sort of the KT event and the sort of wiping out of the dinosaurs, sure, that makes sense. But like, God created them to stamp around and destroy things.
Well, we don't know why. I mean, God likes diversity, right? He created all kinds of different animals.
I don't think God created anything. I think that sort of they evolved over time, and those lineages died. They're gone, right? Sauropods didn't evolve into birds. Theropods did. Like, it's not a case of like, sort of, and adding genetic information, any new sequence of genetic information is a new genetic information. So when you take, for instance, so scales and feathers are essentially the same thing. They're both keratin, right? They're essentially the same thing. And they did an experiment where they managed to turn, by manipulating the DNA, they turned feathers back into scales, right? So they've done experiments on this where they have reverse engineered the transformation of scales to feathers. Okay, so we know that can happen, and that is new information. So a reptile that is a dinosaur, a theropod, getting feathers isn't a big stretch at all. We know that can happen. Like, we've done that ourselves by manipulating the DNA.
This is because stem cells themselves have the ability to do this. Stem cells are a cell
Sure.
that have the information already inside. So they're able to do all of these things like from their built-in function that they're able to do, kind of like those a lot of unicellular organisms, which were supposedly evolved into multicellular. That's because they are using stem cells to do it. That's what the 2019 Nature paper discovered. Before, it was thought that they did this through another process. So stem cells that do it. Stem cells themselves have lots of unique capacity because of what they've already been designed with.
But it doesn't really explain why we're getting the fossil record as we see it. Like, why something that is, sort of chicken size, like Procompsognathus, for instance, like, why is that dinosaur sort of found below birds kind of thing? Like, can you explain that?
I would say because birds could really get outside of a global flood much earlier. I would say that the best example that we could talk about when it would come to a global flood would be the fact that we constantly keep finding the tracks before we ever find the animal itself. That would be much more unique to find. I mean, why would evolution do that? Why would there be fossilized trackways every single time before we get fossil trackways before we actually find the animal itself?
It depends on the location. I mean, the whole point about one of my slides was Tectonic, where we went out and we found the fossil, and then they actually found some tracks earlier than the fossil was dated. So that's a specific example where we found the intermediary fossil before we found. I'm not sure where you get the idea that we always find tracks first. I'm not sure where you get that idea from. But It depends on the area. It depends on whether tracks are easier to form. It depends on a lot of things. Like the problem with fossils is often that they need precise conditions in order to form. At least with sort of pre-mineralization and things like that. But if you're talking about tracks, all that they need is sort of an impressive surface, and that's all they need. So it may just be the case that in certain areas tracks form a lot, but I'm not sure what you're talking about. Like, why do we always find tracks first? I'm not sure what that's supposed to point to.
I would say that the reason why we keep finding tracks before we find the animals is because they're fleeing from a flood. They're moving to the higher ground, and then they're dying and being buried by the flood. That's why. But here's that you said talk about the tetrapod itself that existed approximately. If you go by the fossil record, you find the tracks 20 million years earlier than Tiktaalik, right? Why is that? It's because it's moving away. It's trying to get away from rising floodwaters. The things that can't move are what we find in the deeper layers, right?
Tiktaalik was amphibious. It could breathe underwater and on land, much like the mud skipper or things like that. So fish that can breathe air, or the lungfish, for instance, can breathe air. One of its sort of gill sacs has become a lung. So this isn't like I don't like so what we found is Tiktaalik, and we predicted it within 365 to 385 million years ago, kind of thing. And then we found tracks that were earlier than that. I'm not sure why that would invalidate Tiktaalik at all, because we can't tell whether that organism that made those tracks is more basal or not. We can't tell, because they're just tracks. We can't actually look at it. Morphology, as you sort of pointed out, should take a backseat to genetics, right? And that was what that article was saying. It's like we shouldn't rely on morphology when we can rely on genetics, because genetics is that much more precise. And that sort of, but you sort of termed it as morphology is useless, and it's like, well, no, it's not useless, and that wasn't what this is what I'm saying with the cherry pick articles. That didn't say it was useless. It said that genetics can give us a much better idea of lineages than just morphology, and that's been true forever. I don't know why that would be a surprise. The whole point, the whole takeaway is that genetics can account for the similarities between these creatures, and we can make a phylogenetic tree from those genetic similarities, and it falls perfectly within the nested hierarchy that we sort of predict for it.
I don't know about perfect, because if you genetically put up any other region of the genome, if you test cytochrome B with cytochrome C, you won't get a good nested hierarchy. It isn't there. So they choose cytochrome C, but they ignore other regions. You can look at the Y chromosome, which is used for any paternity testing, or any mitochondria, which is used for paternity testing. And you can go back in time and you don't get the evolutionary tree of life. It's only when you go outside of that into the protein coding regions, which we were predicting, which were functionally neutral, which have first and secondary and third functions. That's what the trees are built off of. So we would say that that's the best. And you like that, and so do I. It's when we get into the fossil record itself that is where we were finding discrepancies. And look, they even say it themselves. Look, there's no consensus in the evolutionary role by these fossil apes. Our overall research found that most stories of human origin are not compatible with the fossils that we find today. And then you can go to another one. Oh, I don't know why I moved that there. Over 150 years of continuous discoveries, information about human origin remains as elusive as owning a debate surrounding their interpretations of fossils. The fossil record bearing the origins.
Yeah, so cytochrome B, that's interesting you bring that up, because that is actually one of the evidences for evolution, that we can trace out how distant these two organisms are apart by seeing, by multiplying their base pair substitutions against cytochrome C more specifically, not cytochrome B. That's the one in mitochondrial DNA. Cytochrome C, we can tell how far things are apart by multiplying their base differences, their base duplications. So if the number of amino acids of the cytochrome C protein in monkeys differ from 1, we can tell that they diverged a certain amount of times ago. So that's actually used for evolution. So I'm not sure what your point is, and the whole point is that these similarities and sort of the mitochondrial DNA, like I'm not sure how you're taking or how you're doing the similarities between creatures, because you're sort of you do know that sort of the current standard is that chimpanzees and humans are sort of 98% roughly similar. Yeah, orangutans are about 97% similar, but it all depends on how you're doing the comparison kind of thing. Right. So I'm not sure. I'm not sure how cytochrome C would actually help you out in this case.
Cytochrome C, the reason we don't really use it too much is for testing the timeline, is because cytochrome C has repairable factors in it. There's recombination that occurs there. We like the Y chromosome and the mitochondria regions themselves because they're non-recombinable. They don't repair themselves. If you see a mutation, it stays there. So that's why when we want to test the date of something, we look in the mitochondria genome and we test backwards. That's why we can land on, oh, this is the ancestor of all humans, because the mitochondria. So if we look in cytochrome C, you're saying, oh, we use that as a mutation rate. Well, it has the ability to repair. So it's a really bad way to date something. It's like testing your muscles of your arm to see how old something is. It can repair itself, so not so good.
Well, sure. But we can tell sort of lineages. We can tell that like sort of pigs, cows, and sheep have like sort of molecule, like they have the same cytokine C molecule, right?
Right. Okay, so basically you're using it to show that the genetic relation goes throughout all life that we can follow. We're saying that those are created different, and they're not mutations. They are allele differences that are nucleotide sequences that have function. And evolution is saying that no, they arose from mutation. We're saying, well, look what happens when a mutation arises. It's detrimental to the organism. So to get 600,
not for long.
Not for long. Well,
So this is what sort of where you're going wrong. That do you think that detrimental mutations accumulate in the genome where they don't? Like even slightly detrimental mutations that doesn't cause the death of the species member. Those detrimental mutations will accumulate and eventually weed out those members of the species who have an accumulation of detrimental mutations. This is the whole point. And that's why that Stanford's model doesn't work when we actually apply it to real life laboratory applications like Drosophila and the Lenski labs and places like that. Those populations should be dead if Stanford was right, but they're not. They're perfectly alive. They're perfectly, being carried out. And the reason why is because mutation rates vary. That's just how it is. Like, I know Stanford doesn't like it. I know his model doesn't account for it, but that's just the way it is. So the mutation rates will vary, and any member of that accumulates too many detrimental mutations will be weeded out of the population, and that's what we actually see happen in those labs in real life, not on his model.
Correct, but John Sanford's model was a prediction on larger mammals like mammoths and walruses and human beings. It was never a prediction on bacteria or fruit flies or yeast. His prediction had nothing to do with those smaller organisms. So that's why it doesn't work on them. His prediction had nothing to do with them. I have visuals somewhere on the computer about that, but that's why it doesn't work on bacteria. That's why it doesn't show up in the Lenski experiment, because they had nothing. There's no bearing. So he predicted that humans would go extinct after about 350 generations. We're around 300, 290, 300. So he said that eventually it would lead up to the problems where humans would be able to no longer reproduce and have problems. And so that was to answer that specific. You might have said something else. I forgot what it was. I'm sorry.
Oh, that's okay. I just mentioned Drosophila and Lenski labs. But okay. You said that doesn't apply to small things, but mutation rates can vary in larger organisms as well. So why would we? And don't get me wrong, like Stanford's model is useful for a set mutation rate over very, very large populations. You can use it for some work, but his idea that humans will go extinct within this time. I'm sorry, just no other geneticists believe that is the case, that detrimental mutations would be weeded out. Now, there is a problem with evolution in humans because due to modern medicine, we have kind of stopped that evolution selection from weeding out detrimental mutations in the human genome. And that's exactly what we see, an accumulation of genetic problems that will get worse, and that has been acknowledged. But that's not outside the scope of evolution. That's just that humans have interrupted the evolutionary process by using medicine to support individuals that ordinarily would not have survived.
Oh, correct. I completely agree. We're getting into gene sequencing and CRISPR-Cas9 where we can clip out mutations and everything like that. We're getting into the
that's not what I mean, Matt. What I mean is like somebody with sort of epilepsy or something, right, may not have survived in the wild kind of thing, but thanks to modern medicine or a blind person, for instance, somebody who was born blind as a genetic defect, we now, because of modern medicine, we can keep them alive. We can help them and things like that. Now, because of that person is still in the gene pool when in the wild they wouldn't have been. So that changes how the dynamics play. There is ways to get out of this, like direct genetic manipulation and exactly the things you just mentioned. There is a way out of this. We can sort of solve these problems by use of genetics, but that's one of the reasons why you're sort of seeing this genetic entropy that's not really entropy. It's just mutations and evolution. There's no such thing as de-evolution. It doesn't exist.
Well, we are seeing lots of different geneticists over time talking about it. They're just trying to come up with solutions, like trying to figure out like Crow's concern and I forget the other guy's name, Kondrashov, why aren't we dead 100 times over? He's trying to figure out, he's thinking maybe selection was stronger in the past and now it's just not. But, basically we're trying to figure out like, how come there's, like you saying, we're outside of selection now, but it's still working outside of nature. We're losing animals all the time from mutations. But
we're not outside of selection. It's just, we've sort of changed the dynamics of it. Selection will always happen, but we've changed the dynamics on it based upon our technologies. That's just a fact. And that's what they're talking about with this sort of less selection kind of thing. And there may be other factors at work that stop animals from, because when you're talking about genetic entropy, you're talking about animals just going extinct because of their deleterious mutations in the wild. We're not talking about sort of, I don't know why we would sort of think that the mutation rates and this genetic entropy wouldn't apply to sort of smaller organisms, but would apply to larger organisms. It's all the same genetic code, and mutation rate, they do vary, but I don't see any reason why the sort of fruit flies or the E. coli should go, thousands of times past when the model says they should have stopped, or become so riddled with deficient mutations that they should stop. Because you can't just say, hey, well, I've got a model for genetics and then not apply it to certain organisms. That's kind of weird, don't you think?
I think because John Sanford noticed that these certain organisms had a very strong, powerful natural selection ability. So they can weed out these deleterious mutations really, really quickly, as selection in human beings and dogs and horses can't do it. Selection's not strong enough, so they keep accumulating over the time. Each successive generation keeps getting these so-called neutral mutations, around 90, and then a few of them are really deleterious, and he's saying that those neutral ones aren't really neutral. They're actually leading to fitness loss as well. Slowly it's accumulating, but that doesn't happen on these smaller organisms. Their selection ability is much stronger. So his predictions are that we'll see it in larger organisms and we'll see their extinction come up real soon. And that's why he made the predictions on the humans. But obviously if that prediction is true, we won't be around to see they come true. We'll just start dying, right?
I mean, there's sort of mammals with shorter lifespans as well, and we don't see that happen in them either. So, I'm not sure why we would think that that's going to happen. And so Sanford doesn't really have any evidence for this. It's just, him saying, hey, humans are going to be dead within a certain amount of time. Well, great. That's a testable prediction. I guess no one's going to be around to test it, but it doesn't seem to play out that way in the experiments that we've done. So, if he could publish, sort of something on that would be great, but he isn't. Like, sort of de-evolution and genetic entropy are not accepted terms anywhere in the field of evolutionary science. They're just not, because that's not what we're talking about. And yes, neutral mutations can crop up. The problem is that they haven't accounted for is that when they do become deleterious due to the overwhelming number of them, that is when the organism is selected against. So there's still a selection mechanism available. I don't really know what else to say there. Maybe we should move on to something else like ERVs or something like that, or chromosome 2 fusion, perhaps.
Sure. Real quick, you said that I said 3 people cannot. There's no way it could lead to all the haplogroups. Very true, except for this is 8 generations later. That's during Peleg's generation that people spread out from the Tower of Babel, and that's when they formed. You said haplogroup A is in Africa, and I don't understand why that's all humans arose from that, and that's why it spread out, and that's why we have them in all people groups.
Yeah.
But I'm saying that that's actually inferred that the haplogroup A is the oldest, and they're the oldest people in Africa. I would agree that haplogroup A isn't people. There's no doubt about that. But that's because Ham's line moved into Africa, and they lived in smaller populations and had more generations than other people. So therefore, they genetically look older. Because if you're going to have twice the amount of generations as the rest of the other people groups, they're going to look twice as old. That was why Nathaniel Jensen predicted that the mutation rate in African people will also be faster, and that we'll also find that they had generation times of a younger one. So that's why haplogroup A, it looks older. It isn't because they're older. It's like testing you and me, and then we go test Amish people. We're going to have more mutations than them because they have a very slow mutation rate because they have kids much later in life.
But haplogroup A did not split into 3, or if the haplogroup A was one of these 3 haplogroups that you said, what are the other 2?
Oh, sorry, no. The 3 haplogroups that were supposedly in Noah's 3 daughter-in-law that spread out was L, M, and N. Those were the 3 main haplogroups, and from them, all other people spread. And it was Ham's that one.
So what was the 3 haplogroups?
L, M, and N.
No, the haplogroups. Well, you're saying that L, M, and N represented these 3 haplogroups. Well, right. What is the name of the genetic haplogroups? Like, is it which was haplogroup A?
Oh, haplogroup A would be considered in the African lineage, but we're just talking about
Okay. I must be misunderstanding, because you said L, M, and N sort of comprised 3 haplogroups, right? They split up, and there's 3 haplogroups, right?
Yes. Yeah.
So if there's 3 haplogroups, one from each of these people, which one formed A?
Oh, good question. Okay, I got you. So we got the 3 daughter-in-law of Noah coming off the ark, and then their descendants go to the Tower of Babel, and then there's a dispersion. Each son inherits 3 different lands. Ham's line inherited Africa. I believe it was in. I'm not positive on that, but his line went over into Africa, and then then the mutations started to occur, and that's where A arose. And that line in particular had more mutations than anything down in South Africa, where it was hot.
Okay. So what were the other 2 haplogroups from this 3 splitting of 3 that you're talking about?
Oh, the L and M. Yeah, that was Nathaniel Jensen's predictions on those are the 3 main haplogroups, which all people groups today descended from. He said they were sisters, and they have a very few mutations between them. So those are the rooted ancestors for humanity. But so
here's the problem. Here's the problem, that we know that there was haplogroup A that split up at the BT split, right? BT as in the one split into B, and then a main group came off of that. The rest came off of that, right? So it split one aside. Then it went to the CT split, which then haplogroup C split off, and then the line kept going, right? And then D and E at the DE split, that split off. Then the main tree kept going with F and continued down. So
Right.
the story that you have told, that three people spread out and they each formed a haplogroup, even though like L and M, I'm presuming, I'm going to guess, because since you can't seem to get there, that's B and C, right? But that wouldn't have happened under the genetics that we see. What we see is a split from A makes B, and the rest kept going. Then a split from C, and then the rest kept going, right? So it isn't... The whole idea that it was sort of a family of three split up just doesn't work here. Hang on, I'll get a sort of thing for you. Hold it. I'll sort of... I'm pretty sure I had a diagram of this.
And gentlemen, we just hit the 33-minute mark. So I just wanted to remind you, I think we have a couple topics on the table still for you both to discuss. So feel free to wrap up this specific topic and we'll move on from there. Matt, feel free to respond to anything to avoid dead air, please.
Okay.
Yeah, sorry. I got it. Sorry. That's my bad. I should have been better prepared.
Sure. Dr. Nathaniel Jensen just used the regular haplogroups that are in the secular literature. And then from there, he said, it's not based on how many mutations there are going back to an ancestor, because mutation rates can differ between people groups. And the smaller population, the more fixed they'll get.
So this is how the haplogroups see. So basically there was a split, and A is still in Africa, right? The rest of the populations traveled out of Africa, but the B population is down towards South Africa, while E is sort of recombined back with populations moving back into Africa. So you've got the BT, where the B split off, and then the T is the rest of the tree kept going. The CT split, D and E split off. So there's a slight difference there. But what I'm trying to point out is genetically, these split off, they weren't three separate individual groups, right? Because we're talking about populations. A segment of the population became distanced from another, enough so we can see the genetic dissimilarities between them. So the whole idea of having three individuals that all sort of found their own area, it doesn't work for how we see the genetics of the haplogroup. It just doesn't.
Well, if you have somebody and they don't have any mutations and they're getting off, and there's and they each have one mutation that's a little bit different. They have great genetics, right? Perfect, no mutations anywhere. They can only create offspring that have new mutations that can arise, so a new haplogroup can form. So if their ancestors go off into a new region and their offspring have a new mutation and it raises to fixation, you get a new haplogroup. So we're saying that haplogroups that arose in Africa were a branch off of one of these other people. And so they created, they reached fixation and they created a new haplogroup. So we're having this diversity and explosion of haplogroups, mostly in the Middle East, not in Africa. So why would we think that it was mostly in Africa? Only because they have more mutations in Africa. We explained that from what I said earlier, is that they have a faster generation time and more generations.
Yeah, I don't see why they would have more mutation rates. To be honest with you, I don't see why that would be the case. I'm not 100% sure. In fact, as I recall, haplogroup A has very low mutation rates from memory.
I think the reason why they believe Africans are older is because they have more mutations than other people groups. Like me and you, we would have a lot less mutations than Africans would. So they believe they're older because each generation is accumulating more mutations. So they have the most than anybody.
Well, Yes, but you've got to differentiate between having the most mutations and mutation rates, right? So
Correct.
they think, yes, so even though the mutation rate might be lower, they can have more. Those things are not that's not conflicting with one another.
Correct. Actually, what evolution believes in neutral theory, which is that you, me, Africans, and everybody mutate at exactly the same rate. Neutral theory poses that we all have about the same mutation rate, let's say 0.5 per generation.
Yeah, roughly. Yeah,
so
sure.
Then we get an Africa. So if that's true, then when you look at Africa, they definitely are older because they have so many more mutations than the rest of the people group. We're saying in the creationist model, no, we're the same age. They've just had more generations, twice the amount as the European people did, because they had offspring around 15 or 16 years of age. So they look a lot older, as where Europeans and Asians have generation times much larger, 30 years of old, 40 years of old. So we don't have as many mutations. So it's based on the assumption that neutral theory is true and that more generations have occurred there. But
well, I think neutral theory sort of says it's roughly the same, but it's not saying exactly the same. Like I'm not sure it is saying what you're claiming here, that the mutation rates for humans are identical. I don't think that's the case. I think compared with other species, yes, it's relatively neutral, but right. I don't think it's exactly the same for everybody. Right. In fact, I've got a paper here that says mutation rate variability across human chromosome Y haplogroups. The earlier we go back towards the original haplogroups, which is B, A, C, the mutation rates get lower.
As we go back in time?
Yes.
Okay. I'm not sure what study that is, but we do have some pretty good stuff on the Y chromosome, but I do like that subject a lot, believe me. But the Y chromosome data is what Nathaniel Jensen got from the mutation rate of Noah only about 4,500 years ago. So he's using a pedigree study. So when you look at a Y chromosome mutation rate that goes back 300 or so thousand years, that's a phylogenetic mutation rate. We don't look at those because those are assuming evolution to be true in the first place. So if you assume that we split from 8 to 7 million years ago, yes, the phylogenetic mutation rate would be accurate. But if you take that away and we only look at the observable rate, which is 3 mutations per generation in the Y chromosome, me and you only have 450 mutations. Well, you only get about 150 generations ago before we reach the Y chromosome ancestor of all human beings. So I don't know how evolution could answer that, but
no.
Have you not heard that before?
No. I mean, chromosome Y, it basically is passed unchanged from father to son. It's basically like, because it doesn't get recombination, it's not mixed with any other DNA, but it does have a high rate of mutation compared to other areas of the genome. So right. I'm not sure where you're getting that number from, because sort of the Y chromosome Adam that we've sort of predicted goes back millions of years. I'm not sure where you got that from.
Oh, we got that. You might not like it, but we got that. Nathaniel Jensen found it. He was looking at pedigree secular papers, and he looked inside of the Y chromosome of a 2018 study, I believe. Matter of fact, here, I'll show screen. That way you can see the study exactly. That way you can talk about it. Sorry.
Yeah, I'm seeing that it basically was 200,000 to 300,000 years ago was sort of the Y chromosome common ancestor.
Yeah, that's right. Okay, here we go. This study, Carmen et al., they ran a mutation rate, and they found that there was a really, really fast mutation rate. The two on the left were pedigree as well, but the two on the left didn't use what's called high sequencing data, which means they didn't scan the Y chromosome more than once. So they didn't find very many mutations. Then they ran it with high sequencing data, and they found a lot of mutations in there. They actually wrote in the supplemental findings, that our findings prompted us to explore additional filters, which means they didn't look at it anymore because the results weren't exactly what they wanted. I have a little cartoon guy, but he flicked it off. But essentially what they did is they scanned it, and they noticed that, oh my gosh, we missed this many mutations if we only scan it one time. So they scanned it.
Can I have that paper, please? Can I grab that paper?
Sure. Yeah, absolutely. Yeah, the Marty et al. Is a good one. Here's the paper, analysis of 62 hybrid assembly of Y chromosome. And what happened is Nathaniel Jensen ran that and found that, wow, each person gets 3 mutations per generation. And he said, if you take that back in time, that's only 4,500 years. So he ran that compared to the evolutionary timeline, and it was completely different. So he said, if there's 3 mutations per generation, and there's 440 in the total in people today, that's just 146 generations ago. So if you use the 30-year generation time, that's about 4,380 years. If you use it 35 years, it goes about 5,000 years.
So that study is called A Recent Bottleneck of Y Chromosome Diversity Coincides with Global Change in Culture. Genetic diversity, but out of Africa dispersal 50 to 100,000 years ago. We detect a cluster of founder haplogroups in a narrow time interval at 47 to 52,000 years ago, consistent with rapid initial colonization model of Eurasia and Oceania after the out of Africa bottleneck. So basically he's saying he found it consistent with the model that said that the bottleneck was caused by cultural changes affecting the variance of reproductive success among males. So basically he's sort of saying, hey, there was a bottleneck. We think it was caused by cultural changes, but nothing about that sort of in any way is against evolution. Like you seem to be cherry picking from these studies to say, hey, they found a problem here, and then they present possible what happened in these problems. But you're missing that. You're just saying, hey, there's a problem, can't be true. That is not what these people are saying, Matt. You need to read the entire study, not just say, hey, they found a problem, evolution untrue. Because he explains what happens. And he explains that it's consistent with a rapid initial colonization model of Eurasia and Oceania, because there are different models. There's a slow progress out of Africa. There's all these different models that they have. And he's saying he supports the one that is rapid colonization along with cultural shifts that meant that there was a bottleneck, not of people, but of who actually got to pass on chromosome Y, right? So if there was a cultural shift and suddenly it was that only the biggest and strongest, sort of got mates and the rest just didn't, then that would create something that we're seeing here. Okay, so it's really important that you actually represent these studies correctly, because this study disagrees with you.
Oh, I have no doubt that it disagrees with us. It disagrees with Jensen too. That's why he said that they hid it in the supplemental area, and they prompted to use different filters because they didn't like what filter C was giving. But that's where they were getting their mutation rate from. So they got results that they didn't like. They used the longer results that showed evolution, because you got to remember, if you and me want grant money for getting a study, we'll get those numbers no matter how.
Oh, look, don't look. I'm not interested in talking about conspiracy theories. There's no conspiracy theory over evolution. There's none. Okay, because you can probably get a lot of money from creation ministries and places like that as well. Probably a better money than you would make just doing sort of evolution research. There's not, like, I don't know why you think there's suddenly this gold mine of money to spend years studying it just to sort of make a scientist's paycheck isn't a good argument. It just isn't. And the fact the fact that there's Christians out there who also believe in evolution and accept evolution as a fact and the theory of evolution, just shows that no, it's not this conspiracy that you think it is. It's just not.
I don't really say conspiracy. I just say you just follow the money. I always challenge anybody to just submit yourself. Write a paper, make sure it's 100% factual. It could be even evolution, I don't care. But just put in there that it's creationist based. They'll reject it immediately. PLOS biology will just instantly reject it. So if that's the type of science that you have to get rid of, and it only has to agree with them, then it's not really conspiracy, because anybody listening can do it. I did it. I was rejected on day one. They didn't actually read it at all. They just said, we're not interested in that material. So how could it ever pass peer review? People go, we want peer reviewed. Well, I'm sorry, I tried. It was rejected immediately. I posted a community post. Here's my rejection letter on from the email.
Yeah. So what experiments did you do? What did you carry out? So you wrote a paper based upon creation.
Correct. But I always tell people you can do it on anything. You write a paper. I did it on linguistics.
Yeah.
Because linguistics is unique. There's about 8 theories on where language arose from. The bow wow theory is the most popular. That man heard an animal and we copied the sound. Well, Eric Lindbergh found that the language can't really arise on its own because nobody over the age of puberty could ever learn language.
So let me guess, you wrote a paper on Tower of Babel, I presume?
Basically, yeah. I wrote a paper.
So that's going to be rejected because creationism is not a science. Okay, there's no science in there. There just isn't. And I'm really sorry that shocks you, Matt, but it just isn't a science. There is no reputable academic facility that's going to take a scientific paper on the Tower of Babel. It just isn't going to happen because the Tower of Babel is a myth. It's just a story. It's barely even history. It's a footnote in history. It's not something that a scientist would take seriously.
But if you don't take it seriously, how could you ever know if it is true? For example, evolution requires a natural explanation for everything. If there's no natural explanation for where language arose from,
yes,
we can give it a good explanation. Why is it rejected without looking at it? Should it not?
Because we only appeal to naturalistic. That is what science is. It's methodological naturalism. That's exactly what it is. There is no spectral evidence in science. You can't say, well, I have a story about a dancing hippopotamus that gave language to the people. It just doesn't get accepted. That is the restrictions of science. And I'm sorry that you don't agree with that, but that is what methodological naturalism is. It doesn't prevent you from getting to a God, but your methodology has to be a natural methodology, not based upon religious stuff, not based upon supernatural things. It has to be a naturalistic explanation because that is the constraint of science. And, because if you accept the same evidence that you have for, sort of languages being handed down by a God, then we've got to accept every explanation from every religion on how languages formed, being gifted by the Hindu gods and things. And then we have contradictory explanations. How could we possibly do that?
Well, we have contradictory explanations in evolution all the time, and people deal with it. There's explanations called the aquatic ape theory, where we evolved from apes that were swimming in the water rather than in the savannah, and that's where we evolved our subcutaneous layer of fat from, rather than living in the plains. That was rejected immediately and considered woo-woo. There's lots of theories that are just outright like, nah, it didn't do good. So there's purely naturalistic papers that get rejected all the time for not being, thorough enough, for not following methodology correctly, there's a million reasons why papers might get rejected. Like, I don't see how you could possibly do any substantive investigation or experimentation on the Tower of Babel, apart from pointing to the Bible. Did you point to the Bible as sort of, one of your evidences for the Tower of Babel and languages? Probably at one point, but
right,
because if it's the history book, we want to point to it at least somewhat. But for evidence,
it's not a history book.
Well,
it's a religious book.
If we test it as a history book,
we don't.
Correct. Well,
we don't point it for a history book.
Well, if we want to test if a history book is true, we got to go to the science. We're doing the same thing with the Bible. That's all. Well,
no, but I mean, yeah, we kind of use sort of evidence in order to test history, but history has its limits. Yeah, you're right. No, Zach's right. We're not really talking about evolution anymore. All I'm saying is that lots of papers get rejected for not having a proper methodology, and it sounds to me like your paper just didn't have a proper methodology based upon methodological naturalism. And that may be a limit of science. Maybe that's a question for the theological sphere, but it certainly isn't a question for the scientific sphere. It just isn't.
Okay, but if we're going to say, that's why I challenge anybody to write up a normal paper and have it have to do with anything on young Earth creation. Let's say you write one right now, and you write one on how dinosaurs evolved into birds, and you say, but from a young Earth creation perspective, it'll just get removed. So it kind of makes it hard for us because you guys want us to be.
What I mean, I really want to steel man your position here. I don't know how I would write that paper because I don't see any naturalistic evidence for things being created. There's just nothing there for me to point to.
It's okay. I mean, I would have there isn't for abiogenesis, but that's still in every scientific textbook. We just go for
no. Hold on. Abiogenesis is in there as a hypothesis, not a theory, right? So the difference is understanding the difference between hypotheses and theory. So we teach evolution because that's a theory. There's a ton of evidence behind it. It basically is as good as a fact in science, basically. They are sure about the theory of evolution as they are about the theory of gravity. Like, they are absolutely sure about it. A hypothesis like abiogenesis, we don't quite understand how it works yet. So abiogenesis might be taught in sort of maybe a college level kind of thing if you're going into genetics, but it will be outlined that we don't know. This is not a theory. It is outlined very, very, very clearly, the difference between something like the theory of evolution and the multiple hypotheses that abiogenesis have. That isn't similar at all.
Well, we still make hypotheses. It's still on the line for us as creationists. So, okay, we can just move the creation model to a hypothesis. We hypothesize that God created everything. That's all. But you said that evolution is a definite fact. Well, why don't we ask you then to give an example, maybe humans. Matter of fact, you can pick an animal too. Where would anything ever be in 10,000 years from now? What could it evolve into? Can you name where anything will be? Because most evolutionists say, well,
I don't know.
Do anything.
It would be pure speculation. It would be pure speculation. So
it has no predictive power, right?
No, well, I mean, just because what? No,
it has no predictive power. You said, I said, pick anything on Earth. Where is it going to evolve? Just in 10,000.
Of course, it does. We can. No. Of course, we can sort of predict change of things, but I don't know the circumstances and the pressures that's going to be applied. I mean, I would guess that things will evolve to, if we continue the rate we're going, that things will evolve to sort of be able to operate with less oxygen and more carbon dioxide in the atmosphere. I would certainly think that animals that cannot adapt to a higher carbon dioxide rate will probably start to die off. But it's very hard to tell, because if we solve that problem, right, if we solve the amount of carbon dioxide that we're putting into the atmosphere, that might never come to pass. That might not be an evolutionary pressure. We've sort of seen, at least sort of behaviorally, animals adapt to humans, but that's in a very, very short time scale. 2,000 years isn't a huge amount, but that is probably one thing I would expect to see should the sort of, carbon dioxide dumping go on.
Okay. All right.
But don't you think that if we dump tons of carbon dioxide into the atmosphere and animals that can't cope with it die off, don't you think we will see animals that have mutated to cope with higher levels of carbon dioxide?
Absolutely. If there was more gravity, they would be much larger. I agree with like adaptation.
That's evolution.
I agree with adaptation.
Well, that's evolution.
Gentlemen, I've been keeping track of topics over the debate. Great job to the both of you so far. I'm really enjoying this, fast-paced and engaging. Correct me if I'm wrong. We've talked the fossil record. We've discussed predictions. We've discussed the nature of science. Do you feel like maybe we should spend the last remaining minutes discussing ERVs and chromosome 2 fusion?
Sure. Yep. Beautiful.
Okay. Yeah. Mark, so you'd like to lead the way?
Mark, considering I didn't answer any of them on the opening,
right. So ERV is probably the strongest evidence out there. So ERV sequences, like specifically HERV and more specifically even HERVK, how did they get into every single cell in the body when we know definitively, 100% sure that they are fragments of viruses?
Well, one would be, are they actually viruses? We like to say in our model that these were created because, remember, according to the evolutionary argument, these were predicted to be junk DNA, completely worthless. It was actually intelligent design that came along and said, no, these aren't junk DNA. Oops, I don't know how I got rid of that. My bad. Again, I would say it comes down to predictive power. Yet again, you like predictions. I want to give them to you. The predictions were from intelligent design that says we predicted that there won't be much junk DNA, that these will be functional DNA elements, and that the genome will be a lot more functional than expected to be. So ERVs ended up being functional units of DNA. So I would say that that was a good success for us. But how can I prove that they were along the lines of what I'm saying as far as inside the genome? Well, again, we go over to predictions. We're saying that,
well, actually, some of them were deleterious. So some of them are cause for diseases. So
yeah,
how do you explain that if they've been created for us? So it seems like you want your cake and eat it too. You want to say, hey, they're going to be these beneficial sequences because they've been created. But it doesn't account for why some of them actually lead to very serious diseases, and how Phoenix Rising, we basically got a virus to reactivate. How did that happen if it's a created sequence?
Good one. One would be viruses have an extremely fast mutation rate. So if they have a faster mutation rate and they're accumulating mutations always cause problems. So it might not just be from the virus. So if the virus is a dead virus and it was inside of us, and then it was completely dead, the mutations alone would be what were causing the disease. So even if it was an ERV that was completely useless and didn't have any function whatsoever, yes, the mutations that are building up in it would be causing strain. Kind of like
I thought you said it wasn't a virus. I thought you said it was a created sequence. So now you're switching.
No, we can say some of them are viruses. Sure, we can't just say humans are completely immune to viruses.
How do you tell which ones? How do you tell which ones are viruses and which ones are created sequences? How do you differentiate between the two? Good
question. Two answers to that. One would be we would look at the entirety of it. Would be the function of it? Are they all functioning? A lot of them do transcription. Since they're doing transcription, we know they require ATP, so that obviously they would be there for a reason, because the body wouldn't waste energy supplying it. Another would be their size. If they're functioning and they're required for the placenta, they have to work, because without them, we don't reproduce anymore. So we need them to live and to have offspring. So when could humans have ever lived without this? Because we couldn't have. So obviously they had to be there from the beginning. That would be another one.
So basically it sounds like that you're saying, hey, if it's beneficial, it's a created sequence, and if it's not, then it's a virus.
Oh, to some degree, yeah, because that was one of the basic tenets of the prediction of junk DNA. We predicted it would have function. So if we look in there and we find the ERV and it has function, we go, clearly that was created. If it doesn't have any function, like you said, and that one retrovirus reactivated and it caused problems, clearly not created.
So that's the Texas sharpshooter fallacy. You're basically taking any case where it has function and saying, see, we were right, and ignoring all of sort of the knockout experiments that they do that show that some of them do not have any function whatsoever. And in fact, some of them have a detrimental effect on humans, and they can be reactivated as viruses, even though they're identical. They're almost identical to all the other sequences in here there that you say is created. So what you're basically doing is just saying, hey, I'm going to pick out the ones that support me and ignore the ones that don't. Just ignore them. Say, hey, they were viruses, all the ones that support me. That is called post hoc rationalization. That isn't a prediction. Like if you sort of say all of them have function, they don't.
Right. Again, yeah, we gotta be real careful, because if we say like Candida, for example, we would say Candida is a very good bacteria created inside of us. It starts to grow too much and we get Candida overgrowth. Now it's detrimental. So we would no longer we wouldn't just change our mind and say, God didn't create it because it's detrimental now. So we're not saying the same thing with ERVs. We're trying to say that the endogenous retroviruses, the functional ones, the very large ones, are all fully functional. Mutations have kind of destroyed them because they have a fast mutation rate, but they're still functional. Same thing with a lot of the other ones. A lot of them do transcriptions. Now, do all of them do it? No, of course not. Like they did a phylogeny test on ERVs with cats, and they found that a cheetah had a baboon ERV inside of it. Well, clearly it wasn't created with that. So it must have eaten a baboon placenta and incorporated it into its own genome. So if we were to test that, would we say that the ERV in the cat wasn't created? Well, of course we would. We would say it was created inside the baboon and got transferred to us. Unfortunately, the virologist would probably be able to tell you much better than I would how to distinguish a created ERV over a non-created one. I even think Donny can answer it better than me, because there's a specific way we can tell based on sequencing, right? So I would have to say that I would just go by the function base, because that was what was predicted. And if it doesn't have a function, we caught it from somewhere else. So it has to look like a gene, and it has to look like a virus, because how else could it protect us from viruses?
So what you're doing is you're essentially going, well, some of these have benefits, like the syncytin gene, for instance. So we're gonna claim that as sort of a created functionality, sort of what God created. And all of the ones that, like the HERVK, for instance, that have sort of variations that cause negative deleterious effects, you're going to label those viruses, even though there's no real way without knowing what they do to tell the difference. They're essentially indistinguishable from one another. The problem here comes that something like syncytin can actually be linked to multiple sclerosis as well. So that may have a deleterious function, even though you're saying it is created. So this is a problem where all you are doing, right, is you are going in and you are finding the specific parts to support your worldview. There is no scientific research to support this. All you're doing is basically saying, I'm going to cherry pick and I'm going to select the ones that have been co-opted, have mutated, so there's a benefit. All the ones that have mutated, so they don't provide a benefit, we're just going to say they're viruses. Well, it's just a story. It's just ad hoc explanation. And some of them do nothing at all. And so that's sort of, the whole junk DNA. Yeah, a lot of them are, do things way more than we thought. Sure. Yeah, absolutely. But the whole idea that sort of, you predicted that there wouldn't be junk DNA, well, there still kind of is. There's sort of inactive regions, there's non-coding regions, there's a whole bunch of stuff that it just doesn't do anything. And we know they don't do anything because we can replace them with other genetic sequences, and the organism is unchanged completely.
Well, I always like to say it would be one thing if it was just a rescue device and I was like, oh, well, God definitely created them. And
it is a rescue device.
But the thing is I don't think you could find a single paper that actually shows a process where a non-functional ERV became functional. So it's not a rescue device, because you can't back up what your theory is, because you just read co-option, but read the paper. They'll never be able to show a non-functional going to function. So
okay, well let me just throw in that where they literally reactivated that Phoenix Rising virus element from a non-functional thing.
But it went functional in a good way, or it went functional in a very bad way? I'm guessing very
well. I mean, when you say very bad, it was a weakened form of the virus, so there may actually be some use for it. So it is fascinating. So when you say a useful way to the organism, yeah, terribly bad, I would say.
We believe that the ERVs are functionally based on a kind. So there's going to be functional ERVs specific. So if I eat a bat, I am going to get a virus that's very deadly for me, but very beneficial for the bat. See the difference?
What is a kind?
Picture a specific species of wolf gets placed on an ark and gets off, and it diversifies. It becomes the family member of all the new species that evolve from that. So it would be a nested hierarchy of a family that came off. That would be a kind, like a wolf kind, and then it diversifies into dog kinds, right? A downward trend, human kind, bat kind. So that's how
bird kind, or is there eagles a kind?
Yeah, eagle would be a kind because it's mentioned in Job, so it must have been on the ark. Ostrich was a kind. It says that God made the ostrich stupid. Pretty ironic.
So the eagles, like wedge-tail eagles and bald American eagles, for instance, they have way different chromosome numbers. They can't interbreed.
Correct. A matter of fact, that's a great way we can try to determine what was on the ark. If I want to determine what horse was on the ark, actually, I would go to the highest chromosome species of the horse, and then it'll probably be in the Middle East. Watch, I guarantee it. It'll be over in Europe, right around that area.
Why highest
because chromosome fusions can occur, and they'll lose the maximum chromosome count. Right? It can only go downhill in our model. So chromosome fusions will be a loss. Right? Well, the maximum would be what would have been on the ark, because then you get off the ark, you can only get chromosome fusion, so we'll get less.
I don't think just chromosome fusions can split, can't they? I mean, well, chromosome 2 fusion, for instance, I mean, that is completely indicative of the evolutionary model. Maybe we should address that quickly.
True. Okay, so let's say that there was a human being that we found today that had more chromosomes. We would go, wow, his lineage, his descendants must have been the first now with a fusion. So he must be the hierarchy of the earliest.
No, because that wouldn't be a human.
Oh, sorry. Well, okay, let's call it whatever we want. Let's say that it was. Do you like the ark analogy? Could I say that there was a wolf that had 60 chromosomes, and then it got off and it speciated into a wolf that now had 50?
Sure. I mean, that's fine. I mean, if things can speciate, things can speciate. That's fine. I don't really have a problem with that. The problem comes that with chromosome 2 fusion, if you had I don't know how you would get a human with more like the pair extra chromosomes, like the 48 chromosomes, right? I don't know.
It was a bad analogy.
You would get that.
I'm sorry. It was a bad analogy. I was just making something up. My bad.
Because sort of, I mean, I suppose I would be completely bewildered as to how that could happen, but it wouldn't be human at that point. You would probably see that split off from the other apes before humans split off.
Well, actually, if the chromosome 2 fusion happened, it had to be in the human line, because if it happened in the ancestor of before the split, both the primates would have it. So there had to be in the human evolutionary line.
Yes.
As a matter of fact, that's why they actually said this. I remember this one. Look, the evidence for a fusion in human chromosome tells you little to nothing about whether or not the humans shared a common ancestor with living apes. The human chromosome fusion argument focuses on a fusion event that is specific to the human line and therefore provides.
What's that a citation from? I notice you don't have any citation there. Who is that from?
It looks like I took it off of a picture of a video. You can see the red line there. I can find the video for you. They usually have citations. I don't know why they don't. I can get that for you.
So that's completely different than what every evolutionary biologist says, and geneticist for that matter, except the few that are sort of in the religious sphere, say about the chromosome 2 fusion.
Yeah, I would imagine so, because they needed the fusion to happen, but they didn't predict it at all. Matter of fact, they predicted humans would have 48, the same as the primate.
Yeah, I think that's just somebody talking. I don't think, you haven't actually given any kind of citation to what paper or what thing that's related to. So I don't really care what people who aren't geneticists and aren't scientists have to say about it.
Well, because Darwin was neither one. He didn't even go through peer review. Everyone believes what Darwin says. We're still going off of Darwin.
Look, this is silly. Darwin was a natural philosopher. He was a naturalist. He didn't have biology degrees and peer review at that point. And he did kind of go through peer review because he did publish a book and it was heavily criticized. And eventually, with the overwhelming amount of evidence, we had to acknowledge that evolution was a thing. He won out because the evidence was solid and the predictions he made came true. So, but the idea that Darwin should have a modern day degree when he lived, back hundreds, that's ridiculous. That's absolutely ridiculous. It's like questioning why Socrates didn't have a degree in philosophy. Come on now, Matt. That's just silly.
Well, they had degrees back then. His just wasn't really for that. But I'm just saying that if we're going to go by, of course, we want to listen to specialists and not some dude just walking down the street on genetics and biology. I agree. I completely agree. Obviously, but when we're talking about a chromosome fusion, we're trying to talk like what makes the best evidence. Like what would have to be the population for a chromosome fusion to occur that could spread to every other human, and all other humans on Earth would have to die. So if they're, okay, you say they're not human because they have extra chromosomes. Okay, so take a Neanderthal. Well, they have the same, but let's take a pre-Neanderthal. What would the population have to be?
Well, so different chromosomal amounts can breed, and sometimes they are sterile and sometimes they're not. Like, for instance, lions and tigers, for instance, the old liger. If it's female, it's still fertile. If it's male, it's sterile. I believe that's how it goes. But a lot of these, like, sometimes they can breed if they're close enough genetically, which those animals or those sort of ancient humans probably would be. So it doesn't need to be like just this split off in the way you're describing. It can be that genetically that fusion was incredibly beneficial for the subjects, and therefore that was passed along. So I don't know why you would sort of go, I mean, we don't know exactly how it happened, sure. But the fact that
I just meant population-wise. It had to have been a very small population, because if you're saying that there's tons of people in the world, well, guess what? There's lots of wolves, but you test wolves, and there's lots of different chromosomes that inside of them. We don't. If there were humans in the past or a different species and there was a chromosome fusion, they should be in the population today, but there's not. There's only humans with the same amount of chromosomes, so they had to be a very small population, don't you think?
No, you're misunderstanding how population genetics works. So you have a beneficial mutation, right? This chromosome 2 fusion, okay, and it provides a benefit for the organisms. So as they procreate, other lineages die off that don't have this benefit. I feel I'm explaining basic evolutionary theory to you. Some of them will be born with the mutation, some of them won't. If it's a beneficial mutation, like it's incredibly beneficial, like all the advantages that humans have, then the ones without that mutation will die off, and the ones with that mutation will remain. And that will occur with some being born with and some being born without over a long period of time, until that mutation is fixed in the population. It becomes a normal part of the population. It doesn't need everybody to have that. It just needs one that confers a huge advantage. And selectively, it's weeded into it's sort of selected and forced into the population over time by the individuals having that mutation from being successful, and the ones not dying, essentially, or failing to reproduce is the other option.
Correct. I'm just saying that for that to happen, these populations would have to be very close. So if a new mutation, like let's say a haplogroup, arises today, how long would it take to spread across? Well, it would be very hard, because the population is huge and diversified everywhere. So the creation model could explain it actually better than the evolutionary model, right? Because we have short bottlenecks, very small population, rapid expansion. Evolution theory has multiple bottlenecks, but down to 10,000 populations, but it lasts for thousands of years, right? So you got a lot of inbreeding problems, and you have a lot of duration for inbreeding to be a problem. And the creationist model has a 1-generation population bottleneck with a massive explosion of the population.
A massive inbreeding problem.
Yeah. Think about the inbreeding problem that would occur from a 10,000-year-old bottleneck with a 10,000 population people. It would be pretty bad.
We see that, but sort of 10,000 population, sort of, as long as the genetic diversity is enough, that's not really a problem.
Exactly. There would have to be expansion and population growth. Unfortunately for the evolution model, that doesn't really happen. The population is small. Even in the Catawba catastrophe, supposedly over 1,000 years, the population was below 10,000. That's a long time to have inbreeding. But for the creationist model, no problem. We have one generation.
It's not necessarily inbreeding, because the way that we select is we select from out groups, not in groups. So cultural and behavioral sort of selection processes can make up for that very, very easily. And among that size of population, there's no problem with inbreeding as long as you're not as long as you're selecting from out groups. It's fine.
What species specifically did this chromosome 2 fusion happen in? Because all we have so far are just humans. We believe Neanderthal, Denisovan, and humans are all the same.
Well, probably sort of possibly an Australopithecine, I would believe.
Okay, so you're a splitter, not a lumper. We're lumpers as creationists. We lump things into categories, right? So we would say that they would be outside of the human lineage. And,
well, see, that's a problem. That's a problem with creation, because there's no problem with not being able to split up, like apes from, the Australopithecine, like Australopithecus afarensis, it is a mosaic. It has traits of both apes and humans. We don't have a problem with that. That's not our problem. That's your problem, because you want to split them into two groups. We don't have to say, hey, we have problems categorizing apes from humans, dogs from cats. We've got the problem splitting them up. Absolutely, we do. But that's what we would expect if it was common ancestry. You have separate groups for both of them. So you're the one with the problem being lumpers, to lump that, because creationists have both lumped afarenses, Australopithecus afarenses, in with apes, and others have lumped it in with humans. So which is it? Is it an ape or a human?
Who predicted this?
Who predicted what?
That
I
they
never
would...
mentioned the prediction.
Oh, okay. I thought you said it was a young Earth creationist that's saying that this is lumped together.
Well, different creationists have lumped in Australopithecines with humans and apes, right? So I'm asking, like, you're a lumper, right? So correct. Do you lump Australopithecus afarenses in with humans or apes?
They would be a different outgroup of an extinct ape species, and the reason why is because of the baraminology. Yeah. So we follow the taxonomic boundary paradox, which is the problem for evolution. It was created by creationists, so that's why we don't have the taxonomic boundary paradox for us, because we're lumpers. We take things and we put them together. We go, the genetic boundaries that I showed you earlier. The genetic boundary paradox.
Okay, I've never heard of these paradoxes that you're saying, and we don't have a problem like we don't have a problem with making boundaries, because as evolution says that everything is related, there's no problem with boundaries, because the boundaries are simply set by us. They're arbitrary. They don't exist. All that happens is we have slow, gradual change across a spectrum from one to the other, and that's what we see in the fossil record. And then we say, hey, at this point, we're going to say it stopped being Australopithecus afarenses and started being the next in the series. Right? So we don't have this problem, because there is no boundary for us. There's no boundary on anything, anywhere.
Correct. But see, taxonomy does have a boundary and evolution does not. So that's what the taxonomic boundary paradox is, because you don't get the convergence, right? So the taxonomic boundary paradox says that there's just lines of independent origins and there's no convergence. So evolution says we need a new model. So that's
a problem. But that's a problem with Linnaean taxonomy. It's not a problem with evolution. It's that we're trying to apply Linnaean taxonomy to something I've described as just a spectrum. So we do categorize things into groups, but that's only for convenience. It doesn't mean there's an actual group there. We've just classified them by certain attributes and characteristics.
Right.
We don't. It's not a problem. It's not a problem for evolution. It's just a problem classifying things.
I see that. Well, that's why they're recreating that classification system. I thought it was a problem. That's why they're remaking it up. But
it's because cladistics is so much more accurate. If we trace things by lineages, we get a way better picture, and that lines up, I might add, with genetics and other things. Because Linnaean taxonomy was made when we only was aware of morphology. We didn't have genetics in order to see where these lineages descended. So we had to do this lumping of things into certain groups kind of thing. But they don't always work.
Right. And you don't think that's peculiar, that those taxonomic boundary paradoxes are exactly what we see in genetics? Why? But it shows the creationist orchard. It shows exactly.
No, it doesn't. No, it doesn't. The Linnaean taxonomy shows the orchard that you're talking about, but like I said, it's very inaccurate to what is actually happening. There is no boundary in evolution, so there is no boundary paradox. It's just
that's what they discovered.
All it's doing is describing how Linnaean taxonomy is very, very terrible at describing evolutionary lineages, because it lumps things into sections that are a slow, constant evolution, rather than just one thing ends here and another thing ends there. Like that isn't what's happening in evolution. It's a constant change.
I agree. But you should see that change in genetics. Shouldn't we see a human going back and converging with a common ancestor of both primitive? That's what was expected. The chimpanzee line, right? A common ancestor, maybe.
Yeah, we do.
Mixing. But when we go back, it lands on nothing but a human. Not long ago. That's what that genetic boundary study showed. Humans came out of a bottleneck. Right?
No, not well. When they say not long ago, they're talking like 100,000, 200,000 years.
Yeah.
Like, so not long ago in geological times is actually a long time, you know, if you're counting on your wristwatch kind of thing. So, but, you know, sort of, so Australopithecus, even though you're sort of saying it's an ape species, it has many of the features that humans have. So when you're saying, hey, shouldn't we see an ancestor that these diverge into? Well, we kind of do.
Okay, well, we got to go back to the predictions. What was made on baraminology regarding these species? They take the shapes of skulls, different morphology, tailbone, the bacillum, the only bone that exists on primates and doesn't in humans, and they were like, okay, let's see if these creatures actually match humans or outside of humans in the primate category. They predicted primate. They line up barometrical distancing and say, look at that. They don't line up, they don't match humans or anything in the human category, Denisovan, Neanderthal, none of that. So what we do is we would say, look at every dog breed on Earth. Let's test the distance between all of their snouts, muzzles, size of their skull, cranium shape, everything. And then put them into a simulation and say, okay, everything within that diversity is related. And then they say, okay, let's test another thing that we say is related to that dog. And then if it has those same features, we know it's related, even if we didn't think that they were. So this is where some people really like it, like McLean or Todd Wood. They're super popular for their baraminology things. And so they're the ones that made predictions on what was human and what was not based on this barometrical distancing, because we don't have genetic data for some of these things.
So what tests, because baraminology? Yeah, that's been criticized to not do any kind of testing or any kind of sort of predictive power whatsoever. You just sort of take what evolution has done and then fit it into the baraminology kind of thing. So how does baraminology show that it is actually a more powerful predictive test than cladistics?
Because what it'll do is when they find something right away, they will lump it or they'll split it. And again, because we want to, as soon as they found, for instance, Neanderthal, what did we do? Some of them were just real quick to say, oh, these are pre-flood humans. Charles Darwin didn't say anything. He was like, wow, it has a really spacious skull, meaning very large and lots of capacity inside of it. He didn't say much else beyond that. So the creationists were like, oh, they must be pre-flood men that lived 900 years old. That's why their sand is so big. And then baraminology said, no, they're just completely human. There's no difference. They just have more morphological differences because they have more diversity in them. The older humans have more diversity. They're dead now, so we lost the diversity. So again, it would go down to the predictive power. So Neanderthal was a complete flop for evolution, as you know. They first predicted that Neanderthal was a non-talking, brutish, ridiculous, covered in hair, furry man that couldn't speak. And then as time went on, they just became more human and more human and more human. And now they're pale people with freckles and red hair. And creationists predicted they were human all along. We were just like, no, they're totally human. Always. They're in the human line. So baraminology, here we go. Found it. Sorry. So
yeah, I don't think that's true. Yeah, so all of these, Australopithecus afarensis, why does it have human elements in it? Why does it have these human elements then?
There'll be some things that just have certain more features than others, because if you start standing up, your spine, like let's say it's circus monkeys, they were required to be on a leash and walk around. So they got lumbar lordosis, where the spine started to curve like an S, like a normal person that was forced to
they did not have the morphology of afarenses. There's no way.
Because
if you're sort of saying that bone distortion due to sort of being in an unnatural position is going to lead up to the morphology of something, that's ridiculous, Matt. Come on now.
No, but it's an example that if they do it over time, if they have to stand, they're going to have a lot of similar features that are indicative of something to stand. Their orbital plane, the foramen magnum, the position is going to change.
Is that what you're claiming for Australopithecus? That it just had the traits of something that stood, not because it was bipedal, but because it just stood up for a while? Is that what you're claiming?
Oh, no, I think it had the ability to stand. That's what was unique about it. It was just a primate that lived in an environment that they walked around. There's animals that walk around all the time. We got kangaroos that walk on two legs. We've got other things that walk around. So it's not the litmus test for being a human. There's animals that walk on two legs. It's not a big deal for us. Again, it comes down to
none with the pelvic socket joint that Australopithecus has that is common with humans.
Oh, that thing. Yeah. We got a guy, C Science, that'll debate you on that one all day long.
C Science can't seem to control himself, so I don't think that will happen.
Oh no! What do you mean, in the comment section?
Oh yeah, last time he was on my channel, he kind of insulted my dead mother, so he's not really welcome back on my channel at all.
Oh my goodness. Well, no good. This Sahelanthropus agensis was predicted to be in the human line. Creationists predicted that it was outside of that. We predicted that it was in baraminology. We said we put it in the human or in the non-human line. We put it way over there. What happens is when we started looking at the different features, we showed that it was they had the features of a female gorilla, and so I don't even have the pictures. My bad. So it got classified as outside of that. And they've said, it's no longer in the human line. They've changed it. It moved on the outside of that now. But that's the first link of the chain of human evolution. So if that's the first link and it's just gone, how about the second link? How about the third? You got to have a complete chain, but it's gone. And there was only one skull to even work with. We're talking about That's why I like it.
So creationists denied Afferences for ages, that it even existed. It was a hoax and things like that. So if you're talking about every prediction that creationists have made, they've said all kinds of things. Now, the morphological characteristics of this particular, what you're calling an ape, sort of the striding bipedal gait, the cranial curvature, the upper limbs and lower spine, they have a convergent great toe. They've got strong arches in their feet. All of this is human things. Like, we would see that and say that is human. That is why we can't differentiate it. Now, you sort of went sideways to something else, like completely, and you didn't address why it has these attributes that is shared by modern humans, not some extinct ape, modern humans.
Well, again, we're going to expect some things to have some similar traits, just like we see animals that have some similar traits. You guys say common ancestry, we say common design. God's going to reuse a lot of features that work. If they work, they work. So if something doesn't climb a tree very often, is it going to need a lot of those features? Probably not. So what was it created for? They're not alive today, so it's very hard to test. We don't have their genetics. So we look at these things, and these are some of the criteria that we have to point to what is in the human line and what is not. Because again, we want morphological differences.
And if it was just one creature, you may have a point. But the problem here comes that we see a long line of Australopithecines with more and more human traits. Like, it's got Australopithecus aminensis, Australopithecus cadaver, Ramidus. We've got sort of all of these Australopithecines. Sediba comes after Africanus, which comes after Australopithecus, and they're all getting more and more human. So your thing is that they would have had to develop more and more human traits, and we have a long line of how the ape became a human from these traits. And you're sort of saying, hey, maybe all of these things developed and then they went extinct. It's ridiculous. It's absolutely ridiculous. They would have to develop the same traits on multiple different occasions just to go extinct. It is a much better explanation that this evolved these traits, and as evolution continued, more and more human traits were evolved. And you're sort of putting an arbitrary barrier in between and saying, hey, these things are humans, these things are apes, while not explaining how these apes had human traits in them, and get more and more human as time progresses, that we can tell from where we find these fossils. As time goes on, becomes more human.
That's good. Go ahead.
Sorry, go ahead.
No, I was just going to say that that's because some of those things are actually classified in the human thing. Like Sediba, for example, that was classified as a human. So very unique, right? That would be classified. You would think that creationists would look at Sediba and say, oh, no, that's a primate.
So,
that's
yeah,
ape.
I noticed that you've got a Homo. It's not Homo Sediba. It's not a Homo genus. It's Australopithecus. It's an Australopithecine.
That's because we classify it as it not being. It's that we believe it's in
yeah.
the Homo.
Well, it's a wrong classification. It's a wrong classification. Then it's Australopithecine.
That's because the fossils are mixed together. It's a mix between the two. So when they took a stick of dynamite and they blew it up in this cave, and it mixed lots of fossils together. That's why it has lots of features. Some features with a primate and some with a human being. It's mixed. We had what's her name? Ann Gibbon or something like that. Donny, I forget her name. She emailed us back and said, "Oh yeah, it's just a mix. It's a conglomeration of two different species." So that makes it really hard. So how do we know when we're looking at two different species with other mixes of different creatures? It's very, very hard. So we make the prediction saying, like, okay, we're going to put this in the human category because it is a human.
Which is completely arbitrary. And the funny thing is that sort of Homo habilis has the attributes of some of these more basal forms like the Australopithecines. That's the thing. You're sort of going, well, we're going to split these things that are basically what you call human but have ape-like traits from the things that are ape-like, that are apes, that you call apes, but they have human-like traits. It seems ridiculous to me that you would have a whole series. We have a vast array of these organisms, these what you call apes with human-like traits, and then as the time progresses, because don't forget stratigraphy, we can tell as we go further down what era that was from, and they start to get into humans with ape-like traits. Like, it's really weird that you would go, okay, we're going to cut it off there, and that's it. And even though Australopithecines, you've got Australopithecine afarenses in one and Australopithecine sediba in another, which is just like, the differences are not that great.
Right. Well, we say that Homo habilis is the junk bin because they throw things into it when they don't know how to classify it or where it goes. So when we say, like, oh, it's got some mixed traits, well, of course, look at the literature. The literature says it's the junk bin. Habilis has all kinds of features. Yeah, it's a junk bin. They throw everything into that.
I'm sure all of the creationist people say that. That is not true at all. Homo habilis is sort of the, like, very far along the trackway to human, but they still do have some ape-like features.
Gentlemen, excellent discussion. We had plenty of bonus minutes worth of discussion, so 50 minutes times 2.
Wow.
But this was a fantastic debate, and it's a comprehensive topic, and so I'm glad that we were able to discuss in detail all of the relevant topics. But just to make sure that we do have time for audience questions, we've only got 34 audience questions. So, gentlemen, we will be done by 4 in the morning. No worries. Okay, Mark and Matt, good job. We ended with smiles. This is good. Smiles all around. So we got closing statements, though. So, gentlemen, I want you guys to bring the heat for 5 more minutes each. Okay, Mark, you get the first 5-minute closing statement. Whenever you're ready, you just let me know, and I'll start your timer. Floor is yours.
Yeah, so a lot of the things I brought up, I don't think Matt really addressed in any kind of sufficiency. A lot of it is sort of post hoc explanations, just changing the story to suit what the biblical narrative is, or what he believes the biblical narrative must express. As there's plenty of Christians that accept evolution and still believe in the Bible. So this whole idea of haplogroups jammed into this sort of after the flood account it doesn't work. It just doesn't work with the way that we see genetics operates. The way that he's describing these DNA sequences as created, even though we've reactivated viruses from these endogenous retroviral elements. It's just basically saying, well, when I need, I think it was kind of ironic that his shirt says rescue device, because it really is a rescue device. You just say, hey, everything that disagrees with my hypothesis is a virus, and everything that agrees, well, that's created. Easy, done. Sure, but that's not science, and it's not scientific in the slightest, and you can believe that, but it seems rather convenient that the deus ex machina allows you to classify things exactly as you need to classify them in order for your sort of ideas to be true. The same was true of the differences between Australopithecines and right up to Homo habilis, Sediba. They want to say, hey, that's a Homo because we have to have it as human, because it is too human to deny. But it also has tons of features that's apes. So which is more likely, that these things arose out of nowhere individually, or that this long series we have of apes getting more and more human-like and losing ape traits is just a long flow of organisms that give birth to one another? Like, you would have to be somehow thinking that someone's fooling you if they created these things to go extinct just to trick us into thinking that there was one long unbroken chain of hominids that came along, and before that Australopithecines. He didn't really address chromosome 2 fusion. We didn't really get much into that, in fairness. But it's interesting that sort of in the rebuttal, he didn't rebut anything that I had to say. And it seems like to me that a lot of this stuff is just, how do we fit it into our genetics into our worldview? A lot of it is cherry picking of papers that vehemently disagree with everything Matt's got to say. It's very easy to pick out problems when the papers say, hey, there is a problem with this, and then it goes on to address it. Well, yeah, there's a problem with all kinds of things. That doesn't mean it's untrue. That is cherry picking. That is simply misrepresenting papers, which is not cool. So all of this kind of stuff, I think I've presented a whole load of very strong evidence, and that's only the tip of the iceberg. There's tons of more evidence for evolution. I kind of recommend a site called BioLogos if you want to investigate it. It's entirely Christians. They're very good. They want to know the facts, and they present the facts. I really highly recommend them if you do want to learn more about evolution, but you don't want to go to a secular website. BioLogos is people of Christianity who can sort of explain all of this stuff. But thank you for listening to me, and I really appreciate being here. Thank you.
Mark, thanks for the 5-minute closing statement. Matt, you also have your 5-minute closing statement, and so let me restart the timer. Whenever you're ready, the floor is yours. Go ahead.
Okay. My opening, I will agree, was a complete failure because I spent my time on testable predictions, nested hierarchy, classification, speciation rates, comparative anatomy, fossil record, why things look more primitive, and mutation rates. I should have spent it on ERVs and chromosome 2 fusion. So that is true. My bad. As I said, I don't debate much, and this is a perfect example of why. So I'm sorry, Mark. Maybe next time. So, but I do hope that you consider looking at that genetic evidence. You will see that that study does show genetic boundaries, exactly what we predicted as creationists. And yes, it does say 200,000 years in the studies, but I want you to realize that those studies aren't looking at a timeline. They're just using assumed phylogenetic mutation rates that assume evolution to be true. They do not use any true observable mutation rate, and those are the ones that are accurate and true. And they're so good because the FBI themselves use them. And if they use the evolutionary rate, they'd be convicting people wrong all the time. The rate's fast. So if a dad is at home and his son goes out and kills somebody and they test the DNA and it looks like it's the dad's, he gets arrested. And then all of a sudden the kid's off free, but then they test it and they find an extra mutation in there. Well, guess what? It was the son that did it, not the dad. So those mutation rates are actually used. They're functional. They're good. They're used in a functional case. So young Earth creation has a mutation rate that's functional. Lots of things are used, and the predictions on them are great. So I feel the evidence against evolution is much stronger than the evidence for it. This is why evolutionists like to talk about the fossil record, right? Like he did tonight. It's subjective. There's no consensus, even in the evolutionary community, on it. It is true that it looks like there's one that exists. We would say that the evidence is clearly from the global flood. Are there going to be discrepancies? Sure, but the discrepancies are for evolution because it's a mess. It's all over the place. It should be, if millions of years are passing, it should be everywhere. There should be index fossils in every single geological layer. Everywhere, you should find it. We don't. So I think the question of everything is, I think the evidence, again, is like the ancient alien concept, right? I think that there's enough evidence for them to make the hypothesis and create a model that ancient aliens did all of these things on Earth. So they clearly have a lot of evidence. Evolution clearly has a lot of what they think are evidence, but I think it's best explained from the creationist model. Now, there's tons of things in the creationist model, all types. And just to say like, oh, well, so what, they predicted that? I mean, that's the gold standard of evolution. It's like of anything, of any model. And genetics is where we shine the most because everything is pointing to very, very recent things. You know, the genetic bottleneck. That wasn't that long ago. And everything came out of it, modern humans as well. So that's what we're saying. And it's not 200,000 years ago. So I also think that another perfect example of how bad evolution is, again, you can ask any evolutionist. I challenge you, ask them to predict where anything is going to be in just 10,000 years. None can give you an answer because they'll admit that evolution can do anything. And that's true, but that proves that it's unpredictable. And if it's unpredictable, that means that you can't make predictions on it. It's that simple. If you can't do it, it's not a very good model, is it? Because our goal is to make predictions on the future. What's good, what's helpful. But if it can't do it, right there, Dr. Nathaniel Jensen's challenges anybody listening right now, pick an animal, it doesn't matter what it is, pick its mutation rate and submit it to AIG. He'll challenge you on it and we'll go. You can go from there. No one's taken him up on it, not even Dr. Dan Cardinale, somebody that he recommended, Creation Myths. He didn't take him up on it. Why not? Because he knows once it's in writing, you're pretty much doomed forever. So I always say, like, put your money where your mouth is. You don't like creation science, that's fine. You can argue against it. But you got to say that if it's making the accurate testable predictions, my guess is that that's where you want to go. Don't just discount it. Is it true that there's some things that we have to take by faith? Absolutely. Matter of fact, one in particular thing it says is that in the beginning, God created the heavens and the earth, right? These are something that we take by faith because we weren't there to witness it. But there was one who did witness it, Jesus. He said he was there. He said in the beginning, he created the male and female. That's what we go by. Therefore, it is a history book. So what flows from that? What can we make predictions on?
1 minute left.
Holy cow, I'm already done. Just don't get emotional about the subject. Just be open-minded. It's fun. It's neat to experience this. I believed in evolution my entire life. I was raised and taught and to believe only in it. I thought it was the only way to go. I even had a book on it. I'm a transhumanist. I believed that I wanted to genetically manipulate my interior and my genetics so that I can't get disease. I was totally fine with it. And then I started investigating another alternative theory, and I was like, "Oh, this is really neat. How unique is that all these things line up with what the Bible's saying is true? It doesn't have to. None of these things had to be true. If we looked in the genetics and we found that there was a human millions of years ago that existed, or a hierarchy or an evolutionary tree of life that can be tested through paternity testing, we're in trouble. All of it we would be in trouble for. But yet but we keep finding the evidence that's validating it. That's why young Earth creation is getting stronger. That's why the population is growing in it. It's amazing.
All right, Matt, thank you for that 5-minute concluding statement. Gentlemen, great job to the both of you. I've got automatic respect for anybody who's willing to engage in the debate octagon. And so I really enjoyed this. You both clearly did a lot of work and prep for this epic debate on evolution. I'm smiling because the live chat tonight has been very lively. It's been very fun and interesting to read and enjoy. And also the debate taking place has also been a lot of fun and engaging. So I will remind the audience, this was, in a way, our main event this month. We've had a ton of events this month. It's been busy. June flew by, but we had about 5 shows a week for the entire month, and we had a lot of great debates on evolution, great debates on age of the earth, soteriology, nature of God, dispensationalism. We had several open mics, and so we are moving into July. And our first July debate is on Monday. It'll be between Snake Was Right and in Howard Stirrup. They'll be debating the topic of dragons. Mark, if
Howard's back. Yeah.
Yeah, you've debated Mark or Howard.
I have, yeah. One of the more confusing debates in my life, yes.
So it should be an interesting one then. You heard it from Mark. So
it should be something.
Well, be here Monday because we'll have our first debate for July on Monday. But next week, we actually have 2 open mics. So we're going to have an open mic on the Transcendental Trinity with Matt Yester. And so skeptics, all welcome to join. That one is Friday, I believe, July 7th. July 6th, we're having our annual, I guess you could say, monthly evolution open mic. And so there we go. 2 open mics for everybody. And then we've got some informal discussions coming up. Matt Slick versus Caleb Percher on the nature of God. Matt Slick and Dan Chapa on limited atonement. So anyways, just a snapshot of the overall material that we have for you. If you're not yet subscribed and you love debates of all kinds, make sure to hit that subscribe button. Okay, gentlemen, break time's over. We're going to get into some of these questions. Mark and Matt, you've given us so much of your time already. And so let me put a timer on. Once we hit between 25 and 30 minutes worth of questions, we'll wrap it up. Mark, it is my understanding you're having an after show. And so, yeah, okay, awesome. People that want to continue the discussion, Mark, Mark, your thumbnail's cracking me up. Good job. I appreciate it. Okay, here we go. Let's get with the first question.
Hey, okay. Man, I like dinosaurs. I think dinosaurs.
Yeah. Hey, listen, I appreciate you're a good sport. Okay, that should be fun though. So people check out Mark's after show to a great debate. I've been looking forward to this. So, okay, first question comes in from Creationist Crybaby. Gentlemen, warnings or disclaimers ahead of time. Got some questions from the opposing side to you, Matt. They're coming at you. They're bringing the heat. And same with you, Mark. We got a lot of creationists. They're coming at you too. So
I wouldn't expect anything less. Yeah.
You're both kind of in the hot seat for this one. I think this is going to be fun. Okay, Creationist Crybaby. Question for Matt. This has to do with the chromosome 2. Where else in the human genome, apart from chromosome 2, do you get head-to-head telomeric repeats in high density in the middle of a chromosome with an additional cryptic centromere? Go ahead, Matt.
Well, one would have to assume that that's a centromere to begin with, but we have the chromosome 9 data, which shows that there is a very much more pristine-looking region for the what looks like a fusion already, with both forward and backwards connecting regions that are chained together. So if we can just find these telomeric repeats that like bind together that look like a fusion in random chromosomes throughout, then who's to say it's the result of a chromosome 2 fusion? If we can find a better looking one in chromosome 9 that's not a fusion, then to me it seems unreasonable to assume that it just was a fusion just because it's needed in the evolutionary idea. There's no reason to say that there had to be a fusion. There only is if you look at it from the perspective of evolution. Creationists are fine with it or not being a fusion.
Okay, Matt, thank you very much. Yeah, Mark, floor is yours. Go
Yeah,
ahead.
yeah. As I explained in my intro, like we wouldn't expect a pristine sequence like that. That is something we would not expect because it has to be degraded in order to make the fusion. Like the idea that sort of, and I think that chromosome 9, I think from memory, it's like 10 repeats. The chromosome 2, so high density, it is not. So that is not an example of high density. The 700 and something that is in chromosome 2 is an example of high density. Generally, telomeres, they do decrease in size the more replication happens, but they can be anywhere but to thousands. But the idea that just because the repeat happens, that is not the prediction that we made, and that is not what we get from chromosome 9.
Thank you very much, Mark. Matt, question was for you. You get the last word.
Okay, I really don't think density is what matters. What matters is the fact that we would be able to find a fusion event. They didn't predict that it would. It was going to be what it was. The prediction wasn't going to be like, oh, we'll find lots of density, but no actual connection. It was the opposite. So I think that's the rescue device, personally. I think that's the exact opposite of what they expected to find, just like they didn't expect there to be no chromosome fusion to begin with. No chromosome differences, I mean.
Okay, gentlemen, thank you for the responses from the both of you. You guys are fun debaters. Okay, so Taylor K. I was going to say, why so serious, to quote a good movie, but you both were smiling there. I like it. I like the positive attitudes. Taylor K. Question for Mark. She's coming at you, Mark. Okay, thank you, Taylor. And she asks, if humans have been evolving for millions of years and in isolated environments, then why don't we see more genetic diversity?
Hasn't been millions of years, hundreds of thousands, maybe. The reason why is because you get a lot of recombination. You get basically groups drifting in and out. You get sharing of genetic information over different populations. So in order for genetic, like sort of speciation, that kind of genetic diversity, and we do get some genetic diversity, obviously sort of the sort of physical traits of different isolated pockets of humans are different. That's how we can sort of, even though race is generally pretty arbitrary, that's how we tell. But A, it has been millions of years, and B, we get sort of movements of other, like sort of, Genghis Khan, for instance, came and invaded Europe and sort of, we get movements of populations around so that the genetic pools do overlap a lot. So that's two of the reasons why we don't see any more genetic diversity. Apart from the genetic diversity we actually do see.
Thank you, Mark. Matt, floor is yours.
I would say that I think that genetic diversity lines up very good with what we see in these pedigree mutation rates. We see a fast mutation rate, and that's why we see not a lot of times passed, because we're seeing a fast mutation rate and only a few hundred generations maximum. So that explains the low genetic diversity that we see in all life. I think with this fast mutation rate that we can see, if a couple 100,000 years had passed, we would see more genetic diversity. So that's me.
Thank you, Matt. Mark, you get the last word. Question.
Yeah, I don't think that sort of genetic that mutation rate, as I've said, like mutation rates can vary significantly, and it doesn't take into account recombination of genetic material and things like that. I don't know where you got the idea that mutation rates are always the same. It's very odd, and the whole idea of that we should see way more genetic diversity over merely a couple of 100,000 years is kind of strange. It sort of suggests that you don't understand what we're talking about when we're talking about the diversity of humans.
Okay, thank you very much, Mark. And next question. Got a few super chats here. Alan, thank you very much for the super chat, the support, and also the question. Question is for Matt. As in the Finch micro change, isn't that the same for people dispersing after the Tower of Babel? It doesn't take that long to have many people groups, question mark.
Those are like phenotypic diversity changes, during actually changes in the season. It actually isn't even the next generation. It can change in the season. Their beaks can change a little bit of the shape. As where human beings, that's more of a generational thing. It takes a little bit longer. You start to get up to the poles, and you're gonna get some shifting around of how the density of the bone and some things like that. But overall, yeah, the morphology and the phenotypic diversity of people is it's pretty good. It's much more than animals. Let's just say that. If you line up all the gorillas or all the chimpanzees and then you compare them with humans have a mass amount of phenotypic differences, as where primates do not. They're very, very similar with one another. You can see them, and you're like, wow, you can't even tell the difference between the species unless you're trained in it. So that would be my answer.
Okay, thank you, Matt. Mark, floor is yours.
You can certainly tell the difference between species of ape. There's no mistaking an orangutan for a chimpanzee. So the whole idea that this micro change, we're talking in sort of when we say not long, we're not talking in 10 years or something, or 20 years or 100 years. We're talking in hundreds of thousands of years. And the micro change in finches may have been something that they were really pressured into. Again, you're making the mistake that mutation rates are always the same no matter what's going on. We know that's untrue. We know that sharks have been largely unchanged for millions of years. We know that there are certain animals that have been largely unchanged for millions of years. Mutation rates are not the same everywhere. Sorry, that's just the case.
Thank you, Mark. Matt, you get the last word. The question was for you.
Okay. I was just going to say that correct. I didn't mean that species between gorilla and orangutan. I can't tell. I meant the species within each family. Like, I couldn't tell you if that's eastern or southern chimpanzee. That's what I meant. And humans definitely stand out. There's much more phenotypic diversity between human beings. And that diversity even rolls over. So obviously Noah on the ark had even more, because there's other humans that are gone now that had even more diversity. So we've been losing the diversity over time, not gaining, which evolution would require.
Okay, thank you, Matt and Mark. Next question is another super chat. Comes in for Mark. Question from Smart Simple Fit. Thank you very much for the support. Mark, the woodpecker has a special balance between its adhesive tongue and solvent throat. Does Mark believe its ancestors continually choked to death until they finally figured out how to swallow?
No, of course not. So like many of these sort of changes, they change slowly. And so it might have been a slightly adhesive tongue and a slightly solvent throat, something that almost had no impact, maybe a very slight benefit. Through selection, that would have been reinforced. Just exactly the same way as you sort of get a big cow and breed it with other big cows, you'll get a bigger and bigger cow until, we have ginormous, sort of things. Just a slight advantage selected for can turn into a large advantage, and that is then selected for and reinforced. So no, you're making the mistake that you think that one of it evolved first, like entirely in the way we see it in modern animals. No, it's a slight change that then is reinforced and reinforced, both systems working with one another, but none were as pronounced as what we have today. Like it's just a question based upon a misunderstanding of how evolution actually works.
Thank you, Mark. Matt, floor is yours. That's pretty much completely geared to him. But I would say that, we believe that there's certain designs and things. So when we hear, for example, the laryngeal nerve, right, a lot of people would say that's a bad design. Just like they might say, like the woodpecker, there's a special balance, but it evolved that. We would say that it was more like created. So sure, the longer the necks distend in a creature, the more problems can arise. There's no doubt about that. But what's better, a longer neck so the animal can reach a food source, or dying because of a laryngeal extension that causes a little bit of problems along the way? I use it as an analogy of underneath the car. The exhaust has to go from why not just put the engine in the back of every car where that doesn't have to travel very far, as opposed to putting it in the front where it can be trouble. well, we like to design certain things, and God likes to design certain things as well. And so there are certain creatures that are designed differently.
Thank you, Matt. Mark, you get the last word. Go ahead.
Well, yeah, I mean, I'll refer to Matt's shirt as a rescue device. God likes to design certain things, sort of bad design rescued out of nowhere. So the laryngeal nerve is an example of a terrible design that, is better fitted by evolution. So I'm glad Matt brought it up. Another is the wisdom teeth, which have essentially no function anymore. They kill us a lot of the time, or at least when we didn't have the technology to take them out. And the eye is an interesting one. Sort of the eye has sort of evolved many times, and we know that these things are not related. Like our eye is worse than the octopus eye, which is a parallel evolution of an eyeball that is vastly superior to our own. It doesn't invert the image, and so it doesn't have a blind spot on it. So the idea that, God likes certain designs, well, apparently not in the case of parallel evolution, but we can explain that very easily by sort of pointing at, hey, eyes are very helpful. If something has sort of the mechanisms like sunspots or eye spots to be able to sense light and darkness, and then that evolves over time, it will evolve an eyeball. But it might not be the same mechanisms or exactly the same mechanisms as something else that evolved an eyeball. So this is sort of a problem for creationists, where they sort of say, hey, God likes certain designs, except when he doesn't. Rescued.
Okay, thank you, Mark and Matt, for both of your responses. Next question comes in from Sunflower. Super chat, thank you very much for the $20 super chat and the support, Sunflower. Question for Matt. At first you said evolution had no predictive power, but when Mark made a prediction based on the CO2, you entertained the possibility but dismissed it as adaptation. Why doesn't that count as a prediction for evolution?
Oh, I was just saying, can we predict what any animal is actually going to become? This is just an arbitrary to say that everything will help to adapt to CO2. I would say that, okay, well, everything will adapt to the rise of temperature on Earth when that increases as well. It's just broad spectrum. So specifics are kind of like more along the line of a predictive power, right? Like genetic similarity. That's very, very specific. We would say we're going to find more genetic similarity in things that look more similar to other things, right? That's more hyper specific, for example. And I like I said, I took it. I took it, but it's like that's very vague and not really what the textbooks are showing. You know what I mean? Like, why not give examples of well, humans are moving to Mars soon, so I would predict that we're going to be able to shrink in body size and eventually look like the gray aliens we see on TV. Okay, there we go. There's some actual prediction. I don't see evolutionists making those in the reports anymore. They make retrodictions. They wait for evidence and then they say, okay, boom, this can be explained from evolution. And so I was hoping that evolutionists would be able to say, now we're going to go against you creationists, and we're going to make head-to-head predictions in the future on specific animals, like Dr. Jensen said. I'll pick a dog, pick a cat, anything in the wild, doesn't matter what it is. We'll make a prediction on what it's going to evolve into, and we'll make a prediction on its mutation rate. Let's do it. But no one takes him up on it.
Okay, thank you, Matt, for your response. Mark, go ahead.
Yeah, I just want to give a big shout out to Sunflower. How you doing, buddy? So that's just because something can't predict everything doesn't mean it has no predictive power. We sort of make these predictions on certain things, but not when we don't have an idea of what to predict. So the thing about evolution is it requires you to know the exact circumstances in order to get that. I mean, it's interesting that Matt predicted that sort of people would grow shorter on Mars, because what we'd actually expect is people to grow taller under less gravity. But that's, sort of an evolutionary prediction the opposite way. Do we know? No, of course we don't. We don't know what pressures there will be on Mars, even sexual selection, right? So if suddenly in the future, crooked noses are attractive to the opposite sex, and that's what everybody selects their partner on, that will become a feature of humankind or any animal that happens. So it's very difficult to make those kind of predictions, but that doesn't mean we haven't made predictions about what we find. like I said, every time we dig down and find the fossil record in the places that we expect, that is a testable prediction. If there is one fossil out of order, if there's a bird in the middle of the the early layers, then that is something that basically falsifies evolution. But that has never happened, not once. So the idea that we can't predict certain things doesn't mean it has no predictive power. And I think that, sort of creationists, I mean, they're predicting the world will end as they have done thousands of times in the past. So, I'm not sure how they could predict anything besides, trumpets blaring and, I don't know, 4 horsemen and things like that. But I mean, I won't hold my breath on that one.
Okay, thank you, Mark. Matt, you get the last word. Question was for you.
Okay. Yeah, I'm glad we don't find any birds in the early fossil record as well, because that would destroy the global flood idea as well. The geologic column is like lots of it's still going to mimic a lot what we would expect from a global flood. As for the college textbooks on evolution, they in the evolutionary textbook Tree Thinking, they don't have a single tree of life that includes the name of a single common ancestor. So they don't even have enough confidence based on the fossil record to even name common ancestors. So there's, like I said, lots of subjective things when it comes to the fossil record. I'm not a big fan of it, obviously. I like genetics and biology and the things that we were talking about today much more.
Okay, thank you for the final word there, Matt. Moving on to the next question. Another super chat comes in from George Bond. Thank you very much for the super chat support. And question for Mark. CO2 during the Cambrian was 7,000 ppm, not 400 of today. Your fossil record shows the most expansion in life. How can you support your assertion?
Yeah, well, I mean, that was specifically for humans. It may be other factors at work. I mean, during the Cambrian explosion, we had an explosion of all types of creatures, not just Thingo. But I mean, I would have to double check that your readings on the Cambrian are true. I presume that you got that from ice core samples. Yeah, so just a quick fact check. It was as high as 4,000 parts per million and 180 during the Quaternary glaciation. So, I mean, there are maybe other factors at work there. I'm not entirely sure what you're on about, and whether there were organisms. How do you tell in that whether organisms were more adapted to carbon dioxide than current organisms? You would have to show that somehow. Good luck, George. Go for it.
Thank you, Mark. Matt, if there's anything you'd like to add, go ahead.
Sure. I think evolution predicted that 4.5 billion years ago, the atmosphere contained almost no oxygen. And then the new research actually came out and showed that right at the very bottom, that actually it was pretty temperate and meteor clear in a watery world, basically a Garden of Eden from the very beginning. So that's kind of, I guess you're right, Mark. I guess he did get it from ice cores. I'm not sure either, though. But yeah, maybe he can update. Oh, wait, there. I was just seeing if he said it in the comment section. I don't know. Right there. Oh, he said you need to show it, Mark. It's your assertion.
Look, I point out, like, somebody asked me one of these questions about what will happen in the future of this. I say it's pure speculation, right? And I'd make this absolutely obvious. It's pure speculation. If CO2 rises, then I would expect, as speculation, to see organisms be able to evolve to cope with that higher CO2 level. Matt agreed with me. So I don't know why you're going back to say, hey, organisms during this time, you're making the assertion that the Cambrian explosion could not have happened because there was a high CO2 level. I don't know that that's true. I don't know that organisms weren't adapted to a higher CO2 level, because you're saying higher CO2 couldn't have had an explosion. you are making that assertion, or at least you're suggesting that assertion. I'm not. Like, you need to actually look at what you're saying, because you're meshing two things that are not necessarily the case. And again, like, just because something was temperate doesn't say anything about the gas levels in that environment. Like, just because it was temperate and balmy doesn't mean there was a ton of oxygen around. In fact, if things are temperate, if they're warmer, it suggests more CO2, not less. So the idea that, well, we predicted there'd be a lot of CO2 in the atmosphere at planetary formation, it's temperate and balmy, therefore no CO2, that's a non sequitur. That does not follow, Matt.
Okay, Mark, thank you very much for the last word on that one. Next one comes in, another super chat from John Q. Smith. Thank you very much. Question for Mark again. Explain why you think matter from nothing is not a miracle. Don't use time and evolution. They have not been proven fact.
No, well, time doesn't exist now. Yeah, no, it's time is just an imagination thing, and evolution is a fact. Sort of, the theory you might disagree with, but things change, heritable characteristics over time. That is a fact, and you've got to learn to deal with that in some way. So the something from nothing is a purview of creationists. Sort of the way that scientists think about it is not creation ex nihilo, from literally nothing. There is simply one book that came out, Lawrence Krauss's book, that was a universe from nothing, that sort of was jumped on to say, oh, you believe in no, they don't. His term for nothing was a quantum field. That is okay. So the simplest way to explain it, if you get matter, and this is a terrible explanation because I'm not a physicist, but if you get matter and antimatter, squish them together, they annihilate. They create a quantum field where there is no gas, no matter, no plasma, nothing of any type. It's just sort of a dispersed energy quantum field. So just imagine if that quantum field split into matter and antimatter. That's a terrible explanation, but it's not nothing, like a philosophical, like I've got nothing in my hand. It's simply a different state. So question is malformed. Time and time's a thing. Like, it's a proven fact that time. I don't. Yeah, terrible question.
Thank you for your response, Mark. Matt, anything you'd like to add?
Yeah, I don't really know about something from nothing, because I'm pretty sure he was what Stephen Hawking said, that there was gravity. I think he mentioned that things came from gravity. Just gravity alone was enough to start everything. So that gravity is still something. It's just not a tangible something. But yeah, I don't know what he meant by the no time. I think he meant time itself hadn't existed yet, because you need time, matter, and space to exist at the same time. Therefore, maybe since they weren't existing, there was no time yet. Therefore, there wasn't evolution yet. So where did everything come from if there was no time? Maybe that's where he was coming from.
Okay.
I'm not
and
sure.
Yeah, that's a good question. Although it's a weird question to say there was no time yet, because if there's no time, how can there be a yet? That's a causal, a temporal statement to say yet. It suggests the passing of time. So you're quickly, sort of dealing with no time. You quickly become into the realms of nonsensical, like, what was there before time? And it's like, well, what does before even mean when there is no time? Because how can there be a before? So Sean Carroll, who's an astrophysicist, he put forward that perhaps in the Big Bang, and we don't know for sure, there is a limitation on how far we can look back with physics, because we get to Planck time and we don't know what's happening past our physics kind of breaks down. But it is possible that start of the universe was stretched out to infinity, that as time, as everything compressed and compressed and compressed, time basically slowed until one moment lasts forever, or one tiny infinitesimal dot lasts forever. Do I know this to be the case? No, I'm not claiming to know, but that is one hypothesis. And if that hypothesis is true, then there is no before time. There is just sort of an eternal universe.
Okay, thank you for the last word, Mark. Another one from John Q. Smith. So we got a John Q. versus Mark Reid impromptu debate here. $5 super chat. Question for Mark. So he says, evolutionists fake many discoveries, claim facts because they write them down. Don't you see you have faith spattered between your facts? Mark, go ahead.
Yeah, something spattered here. Look, they don't fake discoveries. Don't, this whole idea that scientists, this conspiracy theory that scientists are somehow faking it because of the quite frankly not at all huge amounts of money they get is just ridiculous. Claim facts because they don't write them down. I mean, facts are basically what we agree on. So a fact is like, we see the inheritable characteristics of animals change over time. That's a fact. Like, nobody will dispute that. If you breed dogs with heritable characteristics, you can change them over time into another dog. Nobody disagrees with that. That's a fact. What you might disagree with is the way that we model it, what we take from that, how we explain it. And no, none of it relies on faith. I don't know why. Like, it depends on how you're using faith. Some people have different descriptions. Some people think it's, the sort of the confidence in something that's unseen. I may presume that you're using it like that, but no faith required. And the whole idea of faith is you've got to have faith no matter what is presented to you. So maybe I should present it to Matt, like, what would change your opinion of evolution? What would actually change your mind?
Go ahead, Matt.
That's a good one, because I wrote a list up when I was actually watching your debate with McQueen, because I didn't think he addressed your opening statements on those things. So I wrote a nice big old fat list. We'll go over sometime. To answer the question, I don't know if they're necessarily faking it. They're wanting, they'll find something and they'll list the discovery quickly, because that's the sensation. That's the news headlines. Lee Berger's the king of that. He just finds something and goes, new missing link. Is it? We don't know yet. He isn't exactly saying like, I'm going to trick everybody and they're all going to believe this big fat lie. He just knows he's going to present something. It's going to hit the news tabloids, and then later he's going to be like, okay, well, we learned more and we got him now. And that's the main case, I think, that I keep seeing and everything. And then here's one that they do when they find something. This girl admits that the general perception is that there's a competition to get the oldest dates, and there's kudos in it. They know if you find an earlier specimen that's maybe the common ancestor of something, man, you're gonna get something from it. There's gonna be a benefit to you some way or another, whether or not it's publication, the cover of a magazine, whether it's new grant money. There's something to be deemed from it. So that's the main incentive, I think.
Okay, thank you, Matt. Mark, you get the last word. Go ahead.
Yeah, so just because one person says that, and yeah, there's certainly some problems with the scientific method as far as sort of sensationalism is concerned, and it's much less the scientists and more the science reporting magazines that are doing it, that need to get clicks and they need to fund themselves, kind of thing. So you'll often find them over-reporting stuff that the scientists didn't actually say. That they basically say, hey, we found this older specimen, maybe this, and it's reported as, hey, we found the next missing link, which is not the scientist's fault. There definitely is a certain amount of prestige in finding certain things, but these people do have sort of professional integrity. And I'd hope that if you have professional integrity, you would extend to them the same thing. It's not a conspiracy. It's not, and this whole idea means that everybody in the industry is in on it. Basically, is ridiculous, because all of these people cited that Matt cited as sort of people in the evolution sphere, they all still believe in evolution. They still accept it as the truth. So. The only people that disagree with it are people with a vested interest in their religion to disagree with it. That's the only people out there. And that it sort of point out, oh, well, these scientists, they have these interests to gain notoriety and stuff like that. Well, parallel it with the people who have a vested religious fundamentalist incentive for it not to be true, and will basically stop at nothing for it not to be true. They've made a whole bunch of blunders in the past, like sort of saying, hey, this isn't true, denying that Lucy, the Australopithecine afarensis, is actually true, denying a whole bunch isn't true, and they've had to back off on every single claim because, obviously the evidence mounts up and mounts up and mounts up. They gave that, humans and chimpanzees were only 80-something% similar until it showed that if you use that methodology with humans and humans, they only turned out 80% similar. So, there's just, I really think that the creationists have to stop taking pot shots at scientists and really sort of calling into question their integrity.
All right. Thanks, Mark and Matt, for your responses. Next one, another super chat. Thank you very much from CQPO. Appreciate the support. Question for Mark. How can you differentiate an animal with built-in information and an animal gaining new information?
Well, so any new sequence of DNA that's not already in the sequence, like the genome, is new information. So if there is a deletion that occurs, basically the genome loops and it deletes that sequence, it just deletes it out. Technically, that's new information because the sequence is then different. That's how these mutations work. So deletions, repetitions, this is how they work. Like if you're talking about new information, definitely if you get a retrovirus insert itself into your genome, that is definitely 100% new information that's come from outside of you completely. It may not be good information, but then it can mutate and become neutral. It doesn't do anything. That's definitely new information. It can be co-opted, so it can mutate and actually be beneficial, and that is new information. So this whole idea of new information, it's like, because there's only 4 sort of letters, TACG in DNA, it's kind of like saying, well, those already exist. Well, yeah, that's like saying, well, if I make up a word with letters, well, if the letters already exist, then it's not a new word. Is ridiculous. Of course it's a new word because it's a different sequence. So I think gaining sort of an animal built-in information would be the way that sort of epigenetics happens, the way that the genome is expressed, that the sequence doesn't in fact change, but regions are turned on and off. That would be built-in information as far as I would consider it.
Okay, thank you, Mark. Again, thanks for the support from Christians questioning popular opinion. Matt, floor is yours.
It's always hard to answer it when the question is definitely geared to somebody else. But I would say that differentiating between built-in information, he mentioned ERVs, would be the difference between a fixed ERV, we would say created, unfixed, obviously assimilated into the genome at one point and now causes problems. So created versus non, right? So same thing with mutations. We look over and a mutation, boom, changes the sequence. That now how it's read, and it's going to express something different. So that information is now changed, and we would say it's oftentimes going to be for the worse. And the information that comes from mutation can't lead to the amount of evolution that's required in the textbooks in the linear scale of things. That's why we don't see it in experiments from really small bacteria, really living short amounts of time, to the point where they become something else. And in laboratories, we can induce them with radiation, feed them anything we want, control the temperature, and regulate supposed 100 million years within months of time. And we just don't see those things. So information from our perspective is just different.
Thank you, Matt. Mark, you get the last word. Go ahead.
Yeah, that's kind of weird because sort of, we don't see them fixed. Of course they're fixed. Sort of these viral sequences have to be fixed to be part of the normal human genome. So sort of saying they're unfixed means that they're not part of the standard. Like it's very weird for you to say so, because we know that, like the HRV, K that I expressed, that was the Phoenix Rising, that is a fixed part of the genome. Like, I don't know why you would say that. And sort of to say we haven't seen sort of these very fast replicating things turn into anything else. Of course we have. There's a Cladomonae reinhardti, which is the algae that became multicellular. Of course we see massive changes in sort of very short-lived or short cycle organisms, as well as, you know, things like in the Lenski labs doing the E. coli experiments on citric acid. Definitely we see sort of changes. It's just that it takes longer for the changes that you're sort of talking about to actually occur. But I think the jump from single cell to multicellularity is a huge leap, evolutionarily speaking, that you know, forward kind of thing. So I'm not sure where you get that information from, that we don't see these changes, because we do.
Okay, thank you for the responses, Mark and Matt. And we've made it to the end. We got one last question. One of your biggest fans, Mark. Question from Bubblegum Gun. $2 super chat. Thank you very much. Short and sweet. Mark, how long ago did the HERV-K infect the genome?
Yeah, I would have to look it up. I think it was when humans split from chimpanzees and stuff, but I would certainly have to look it up. I think from memory about a few million years ago, I believe, but I would have to look it up. And just I don't have that information. Okay, so the youngest, if you're talking about the youngest that I was talking about, the HML2 Pro virus integrated as recently as 100,000 years ago. So relatively recently. Okay,
appreciate it, Mark. Matt, floor is yours. Any thoughts?
Well, HERVQ would be the human line. So obviously we would say at creation. But if it was inserted, yes, it would have been inserted fairly soon after creation, probably went in. But if it went in, because again, I think HERVK has something to do with the placental development. So I think humans require it. Again, I would have to say then probably at creation. It's a functional thing required for reproduction, as far as I remember, the HERVK. Yeah, I'll just go with that.
Okay, thanks, Matt. Mark, you get the last word. Go ahead.
Yeah, so it's syncytin is the placental one, so a different family. The HERVK is responsible for something else, but this particular one, so 100,000 years ago. So really, if you're talking sort of 6,000 to 10,000 years ago, I sincerely doubt that's enough time for the retrovirus to insert, be endogenized, so passed vertically down through generations, and then be fixed in the population, so it becomes a normal part of the human genome in that amount of time, and of course mutate, so it no longer is an infectious agent, because don't forget, without manipulation, the HERVK does not infect. We have to manipulate it slightly, very, very slightly, but slightly, in order to turn that back into an infectious agent, which we can do. So all of that within a few thousand years, the mutation rates must be insane, just insane.
Okay, Mark, thank you for the final word, and that wraps up the audience Q&A. It's been a comprehensive debate. It's been a long debate, and so I want to thank our guests tonight, Mark Reid and Matt, for the time that they've given to us for this much anticipated debate. I've looked forward to it. The audience has, and I think you guys lived up to the hype. It's been a great debate. Lots of great topics and points discussed. This was enjoyable. I look forward to re-listening to this. Okay, so with that, let's get some final words, quick final words and thoughts before we call it a night. And Mark, I know you got an after show to get to. So therefore, Mark, let's start with you. Thanks again for doing this. Lots of fun as usual. Final words, final thoughts.
Oh, my pleasure. I love coming in and debating. It's really good fun. Thanks to Matt for being here and providing opposition, and thanks Donny for moderating, and thank you everybody for listening in and sort of giving me a fair shot. I think that all in all, I think that sort of we've got sort of more evidence for evolution than just about anything else. I find it hard to, because I'm really going back to Matt didn't really answer what would convince him so much. All that would convince me is a reasonable amount of evidence that falsifies evolution. That's it. So sort of like I said, finding a fossil out of place where it should be there, finding something that doesn't fit within the evolutionary tree, or we can't fit within the evolutionary tree would do it. Just finding something out of the ordinary, very, very, small amount of evidence, but compelling, overwhelming, and reliably gathered that shows that evolution is false would do it for me.
Okay, Mark, thank you for the final words and final thoughts. Matt, thank you as well for nearly 3.5 hours of your time for this awesome debate. So quick final words, final thoughts.
Yeah, I had a good time. I don't debate much just because I'm not good at it, but I like Mark. That was interesting. I was nervous. I was training a lot for this one too. I didn't know what was going on. I prepared like 1,000 photos and then I lost them right at the beginning. I was like, oh no, I don't know what I'm going to do. But that was fun. It was good. I just try to answer everything. And I've done that in my last debates. And this one was a disaster because he wanted 2 topics in particular. I didn't really get to them. So I'm sorry, Mark.
No, that's okay. That's right. No. I only meant in your rebuttal kind of thing. It's perfectly fine. It's not a problem. It's just, that's usually what the rebuttal's for. It's more if you want to waste your rebuttal, go for it. It's not it doesn't worry me. No
big deal. A long debate though is pretty long. But I'm fine with it. Overall, good. I guess I won't be seeing you at the after show. This is too much.
No, that's fine. That's
hours,
fine.
I'm good, man.
Well, I mean, everyone's welcome as long as we're sort of respectful, and I promise to be as well. So, that's fine. It's not a problem.
Cool. Good.
All right, gentlemen. I think you guys played off each other well. This was a fun debate. Definitely no boring parts. You guys kept our attention, and so I want to thank you for that. Mark, Matt, again, thank you for your time. To the audience, thank you for tuning in. We've had, over 100 people the entire time for 3.5 hours. So, Mark, Matt, thank you. To the audience, thanks for your questions, support, and we'll see you on Monday. Standing for Truth is out. God bless all.